Good morning. My name is Archana Chatterjee. I'm the Dean of the Chicago Medical School and Vice President for Medical Affairs at Rosalind Franklin University of Medicine and Science. I'm a pediatric infectious diseases specialist by background and have an interest in...
Thank you. Dr. Cody Meissner. Dr. Meissner?
Thank you. I'm Cody Meissner. I'm a professor of pediatrics in the infectious disease division at Tufts University School of Medicine and Tufts Children's Hospital in Boston.
Great. Dr. Gans.
Good morning. This is Dr. Haley Gams. I'm a professor of pediatrics at the Pediatric Infectious Diseases at Stanford University. It's great to be here.
Thank you. Dr. Kurila, Mike Kurila.
Good morning. Michael Kurila. I'm the director of the Division of Clinical Innovation at the National Center for Advancing Translational Science within the National Institute of Health. I'm a pathologist by training, and most of my professional career has been involved in infectious disease drug and vaccine development.
Great. Dr. Paul Offit.
Hi, I'm Paul Offit. I am a professor of pediatrics in the Division of Infectious Diseases at the Children's Hospital of Philadelphia and the Perelman School of Medicine at the University of Pennsylvania.
Dr. Annunziato, Paula Annunziato.
Good morning. I'm Paula Annunziato. I lead clinical global development for vaccines at Merck, and I'm here today as the non-voting industry representative.
Excellent. Mr. Sheldon Tobman. Mr. Tobman. Okay, Dr. Steve Pergam.
Good morning. Can you hear me?
Yes, we can.
Thank you.
Yes, thank you.
Yeah, good morning. My name is Sheldon Taubman. I'm an attorney. I represent low-income clients mostly in the health area. I'm employed by New Haven Legal Assistance Association, although I'm here today in my personal capacity as a consumer representative.
Great. Dr. Steve Pergam.
Hi, everyone. I'm Steve Pergam. I'm an associate professor at the University of Washington and Fred Hutchinson Cancer Research Center, and I focus on infectious diseases.
Mike, can we go to the next slide, please? Great. Dr. Fuller, Ovita Fuller.
I'm Ovita Fuller. Good morning. I'm Ovita Fuller. I'm an associate professor of microbiology and immunology at the University of Michigan Medical School and a member of the African Studies Center in the International Institute, and I'm a virologist by training.
Great. Dr. Kim, Captain Kim.
Good morning, David Kim. I'm the Director of the Division of Vaccines at the Office of Infectious Disease and HIV/AIDS Policy under the Office of the Assistant Secretary for Health.
Great. Dr. Eric Rubin.
Hi, I'm Eric Rubin. I'm editor-in-chief of the New England Journal of Medicine, a professor at the Harvard T.H. Chan School of Public Health, and an infectious disease clinician at the Brigham and Women's Hospital.
Excellent. Dr. James Hildreth.
Good morning. I'm James Hildreth. I'm the president and CEO of Meharry Medical College, and professor of internal medicine. I'm a viral immunologist by training. Thank you.
Great. Dr. Janet Lee.
Good morning. I'm Jeanette Lee. I'm a professor of biostatistics at the University of Arkansas for Medical Sciences.
Great. Next slide, please. Dr. Juan Banaclo.
Good morning. I'm Juan G. Abanacloche. I'm an infectious diseases clinician at the Mayo Clinic in Phoenix, Arizona.
Great. Dr. Mark Sawyer.
Good morning. I'm Mark Sawyer. I'm a professor of pediatrics at the University of California, San Diego, and radiology in San Diego, and I'm a pediatric infectious disease specialist.
Excellent. Dr. Melinda Watson.
Good morning. I'm Melinda Horton. I'm an adult infectious disease physician by training, and I'm director of the Immunization Services Division at the Centers for Disease Control and Prevention.
Great. Thank you. Dr. Ofer Levy.
Good morning. My name is Ofer Levy. I'm the director of the Precision Vaccines Program at Boston Children's Hospital and professor of pediatrics at Harvard Medical School.
Great. Dr. Pamela McInnes.
I'm Pamela McInnes, retired Deputy Director of the National Center for Advancing Translational Sciences at the National Institutes of Health. Good morning.
Great. Dr. Patrick Moore.
Good morning. I'm Patrick Moore. I'm at the University of Pittsburgh Cancer Institute and the Department of Microbiology and Molecular Genetics.
Great. Dr. Ralph Tripp.
Good morning. I'm Ralph Tripp from the University of Georgia. I'm the chair of vaccine therapeutics there.
Okay. Dr. Stanley Perlman. We can't hear you, Dr. Perlman. Mike, can you adjust the volume?
Yes, good morning. I am Dr. Stanley Perlman from the University of Iowa in the Department of Microbiology and Immunology and in pediatric infectious diseases, and I'm a long-term coronavirologist.
Great. I think Dr. Amanda Cohn is available. Can she introduce herself just for a second? We can't hear you.
Can you hear me now?
Yes.
Good morning. Sorry for the technical difficulties. I'm Captain Amanda Cohn, a Chief Medical Officer of the National Center for Immunization and Respiratory Diseases, and a pediatrician by training with expertise in vaccines and pediatrics.
Thank you so much. Okay, let's now introduce introductions of the FDA staff. First, I would like to introduce Dr. Peter Marks, the head of the Center for Biologics. Dr. Marks.
Hi, so thanks very much. Peter Marks, Director, Center for Biologics Evaluation and Research, and I just want to take a moment to thank all of the committee members as well as everybody who's tuning in right now who might not usually be viewing our usually sedate vaccines and related biologics products advisory committee meeting. Thanks very much.
Excellent. Let's move on to Dr. Marianne Gruber, Director of the Office of the Vaccines at CBER.
Yeah, good morning. My name is Marion Gruber, and I'm the Director of the Office of Vaccines Research and Review in the Center for Biologics and Research at FDA. And on behalf of my colleagues in the Office of Vaccines, I also would like to welcome the committee members, Pfizer-BioNTech, as well as the public to today's meeting. I would like to take the opportunity to thank the members of this committee for taking time out of their busy schedule to provide their perspectives, their recommendations, and advice regarding the adequacy of the scientific evidence that will be presented by Pfizer and the FDA today to support a determination whether the benefits of Pfizer-BioNTech's COVID-19 vaccine outweigh its risks. to support authorization of this product under an EUA. FDA very much appreciates the committee's input on this very important topic, and I look forward to today's discussion. Thank you.
Great. Thank you, Dr. Gruber. Now I would like to acknowledge the presence of Dr. Celia Witten, Deputy Director of CBOR, and also Dr. Philip Krause, Deputy Director of Office of Vaccines, who may join later making remarks. So I will now proceed with the conflict of interest statement. Okay, so the Food and Drug Administration is convening virtually today, December 10th, 2020, the 162nd meeting of the Vaccines and Related Biological Products Advisory Committee under the authority of the Federal Advisory Committee Act of 1972. Dr. Arnold Monto is serving as the acting voting chair for this meeting. Today on December 10th, 2020, the committee will meet in open session to discuss the emergency use authorization, EUA, of the Pfizer-BioNTech COVID-19 vaccine for the prevention of COVID-19 in individuals 16 years of age and older. The topic is determined to be a particular matter involving specific parties. With the exception of industry representative member, all standing and temporary voting members of the VERPAC are appointed special government employees or regular government employees from other agencies and are subjected to federal conflicts of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws, including but not limited to 18 United States Code Section 208, is being provided to participants in today's meeting and to the public. Related to the discussions at this meeting, all members, regular government employees and special government employee consultants of this committee have been thoroughly screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouse or minor children, and for the purpose of 18 U.S. Code 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts and grants, cooperative research and development agreements or CRADAs, teaching, speaking, writing, patents and royalties, and primary employment. These may include interests that are current or under negotiation. FDA has determined that all members of this advisory committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized the FDA to grant waivers to special government employees and regular government employees who have financial conflicts of interest when it is determined that the agency's need for a special government employee services outweighs the potential for a conflict of interest created by the financial interest involved, or when the interest of the regular government employee is not so substantial as to be deemed likely to affect the integrity of the services with which the government may expect from the employee. We have the following consultants serving as temporary voting members today. They are Dr. Ovita Fuller, Dr. Juan Benaclock, Dr. James Hildreth, Captain David Kim, Janet Lee, Dr. Ofer Levy, Dr. Pamela McInnes, Patrick Moore, Dr. Stanley Perlman, Eric Rubin, Mark Sawyer, and Ralph Tripp, and Melinda Wharton. Based on today's agenda and all financial interests reported by committee members and consultants, there has been only one conflict of interest waiver issued under 18 U.S. Code 208 in connection with this meeting. Among these consultants, Dr. James Hildreth, a special government employee, has been issued a waiver for his participation in today's meeting. The waiver was already posted on FDA website for public disclosure. Dr. Paula Annunziato is currently serving as the industry representative to this committee. Dr. Annunziato is employed by Merck. Industry representatives are not appointed as special government employees and serve as non-voting members of the committee. Industry representative on this committee is not screened for their financial interests. Industry representatives act on behalf of all regulated industry and bring general industry perspective to the committee. Mr. Sheldon Taubman is serving as the consumer representative for this committee. Consumer representatives are appointed special government employees and are screened and cleared prior to their participation in the meeting. They are voting members of the committee. Today's meeting has multiple external speakers. We have 3 speakers from the Centers for Disease Control and Prevention. These are Dr. Nancy Masonia, Dr. Aaron Hall, Dr. Anita Patel. Regular government employees have all been screened for conflicts of interest and have been cleared to participate as speakers for today's meeting. The guest speaker for this meeting is Dr. Stephen Goodman, a professor of medicine and associate dean for clinical and translational research at Stanford University. He has been asked to disclose any financial interest he may have related to the product before the meeting. Disclosure of conflicts of interest for guest speakers follows all applicable federal laws, regulations, and FDA guidance. FDA encourages all meeting participants, including open public hearing speakers, to advise the committee of any financial relationships that they may have with any affected firm, its products, and if known, its direct competitors. We would like to remind standing and temporary members today that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to inform the DFO and exclude themselves from such involvement, and their exclusion will be noted for the record. This concludes my reading of the conflicts of interest statement for the public record. At this time, I would like to hand over the meeting to our chair, Dr. Manto. Thank you very much. Dr. Manto?
Thank you, Prabha. Here I am. Technical issues, which are going to be one of our biggest problems, I predict, during the day. Thank you for getting the meeting kicked off. I'd like first, and we're actually running a little early, to call on Dr. Doron Singh of the FDA to talk about the situation that we are facing and the presentation of a description of the emergency use authorization. Dr. Fink.
Good morning. I'm Doran Fink. I'm the Deputy Director for Clinical Review in the Division of Vaccines and Related Products Applications within the Office of Vaccines Research and Review, Center for Biologics Evaluation and Research at FDA. The VRBPAC last convened on October 22nd of this year to discuss the development, licensure, and emergency use authorization of COVID-19 preventive vaccines. Since that meeting, COVID cases and associated hospitalizations and deaths have increased substantially in the U.S. and worldwide. On November 20th, Pfizer submitted an emergency use authorization request for the Pfizer-BioNTech COVID-19 vaccine, otherwise known as BNT162b2. This is an mRNA and lipid nanoparticle vaccine administered as a 2-dose regimen 21 days apart. We'll be hearing more about the vaccine and its proposed use in later presentations. The use being requested for emergency authorization is for active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 16 years of age and older. The information submitted with the request includes safety and efficacy data from a large, randomized, blinded, placebo-controlled phase 3 trial. And these data will be discussed in detail in our afternoon sessions. Today, we will be considering whether to make available to millions of Americans an as-yet investigational vaccine that has been developed, tested, and reviewed in record time, with additional testing still underway in ongoing studies. The American public demands and deserves a rigorous, comprehensive, and independent review of the data. And that's what FDA physicians and scientists, all of us career public health servants, have been doing over days, nights, weekends, and yes, over the Thanksgiving holiday. This is in addition to months of review work already completed on information previously submitted in preparation for an EUA request. FDA has been conducting its comprehensive review of the Pfizer-BioNTech COVID-19 vaccine EUA submission since its submission on November 20th. Our review has included verification of clinical data integrity, of Pfizer analyses, and also our own independent analyses using data sets that were provided in the submission. We have continued an ongoing review of chemistry, manufacturing, and control information, non-clinical data, and review of clinical assays, including information that was submitted shortly prior to the EUA request. We have been reviewing and revising, along with Pfizer, prescribing information and fact sheets for vaccine recipients and healthcare providers. These will be necessary to inform and instruct vaccine recipients and healthcare providers during use of the vaccine under EUA, and these materials are necessarily informed by our review of the data. We have sent and received back answers to multiple information requests addressed to Pfizer to clarify questions related to the data. And last but certainly not least, we have been preparing for today's VRBPAC meeting. Today's meeting, in which the committee will advise FDA with its own independent assessment of the data, continues FDA's commitment to an expedited review process that is transparent, scientifically sound, and data-driven. The legal authority for emergency use authorization was established in Section 564 of the Federal Food, Drug, and Cosmetic Act. This legal authority allows for FDA authorization of unapproved medical products or unapproved uses of approved medical products to address public health emergencies related to biological, chemical, radiological, or nuclear agents. Issuance of an emergency use authorization requires prior determination of a threat and declaration of circumstances justifying the need for an EUA to address that threat by the Secretary of Homeland Security, Defense, or Health and Human Services. To that end, Health and Human Services Secretary Azar issued a declaration on March 27th of this year justifying emergency use authorization of drugs and biological products to address the COVID-19 pandemic. Once that declaration has been issued, there are 4 criteria that must be met in order to issue an EUA. First of all, the agent referred to in the EUA declaration can cause a serious or life-threatening disease or condition. We know this to be true for SARS coronavirus 2 and COVID-19. The second and third criteria are closely linked. There must be a reason to believe that the medical product may be effective to prevent, diagnose, or treat the serious or life-threatening condition caused by the agent, and the known and potential benefits of the product should outweigh the known and potential risks of the product. There are special considerations for a COVID-19 vaccine anticipated for widespread deployment to millions of individuals, and these will be discussed on my next slides. The final criterion is that there should be no adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. At this time, the only FDA-approved product for COVID-19 is remdesivir. This is an antiviral agent that is approved for treatment of COVID-19, not prevention. Additional products have been issued under emergency use authorization but have not been FDA approved, and none of these products is authorized for use to prevent COVID-19. Thus, at this time, there is no adequate, approved, and available alternative to a COVID-19 vaccine for preventing COVID-19 caused by SARS coronavirus 2. In October of this year, FDA released guidance outlining our expectations for submissions requesting emergency use authorization of COVID-19 vaccines. And these expectations were discussed at the VRBPAC meeting in October. There are three main areas covered by our expectations. First, data to demonstrate manufacturing quality and consistency. FDA has reviewed the manufacturing quality and consistency data for the Pfizer-BioNTech vaccine and found it adequate to support emergency use authorization of the vaccine. This will not be discussed in detail further at this meeting. Second, we expect clear and compelling safety and efficacy data to support a favorable benefit-risk of the vaccine when rapidly deployed for administration to millions of individuals, including healthy people. And finally, we expect plans for further evaluation of the vaccine safety and effectiveness, including in ongoing clinical trials, active and passive safety monitoring during their use under EUA, as well as observational studies. In terms of clinical data expected to support an EUA submission for a COVID-19 vaccine, we expect a high bar for efficacy. Efficacy data from at least one well-designed phase 3 trial to demonstrate protection against SARS-CoV-2 infection or disease with a point estimate of at least 50% compared to a placebo. Additionally, the appropriately alpha-adjusted confidence interval lower bound around that point estimate should be greater than 30%. This is to ensure that a widely deployed COVID-19 vaccine will have an appreciable impact. In terms of safety data, we expect these data from throughout clinical development to evaluate reactogenicity, serious adverse events, and adverse events of special interest. And we expect that a high proportion of phase 3 study subjects will have been followed for at least 1 month after completion of the full vaccination regimen. We have an additional expectation for follow-up that I will explain on my next slide. We also expect to be able to review sufficient cases of severe COVID-19 that have occurred in clinical trial participants to assess for signals of enhanced disease, and also, if possible, to assess for preliminary evidence of protection against severe disease. We recognize that a planned case-driven interim efficacy analysis and associated safety analyses at the same time could provide data to support an emergency use authorization. We have explained that we expect these analyses to include a median follow-up duration of at least 2 months after completion of the full vaccination regimen. The reasons for this expectation are that, first of all, it allows time for potential immune-mediated adverse events to be evaluated, understanding that uncommon but clinically significant immune-mediated adverse events to preventive vaccines generally have onset within the first 6 weeks following vaccination. A median follow-up of 2 months also ensures that vaccine efficacy is assessed during the time when adaptive and/or memory immune responses, rather than innate responses, are mediating protection. And finally, this follow-up period allows for early assessment of waning protection and for assessment of signals of enhanced disease. Following issuance of an EUA for a COVID-19 vaccine, we understand and expect that further vaccine evaluation would be needed for ongoing benefit-risk assessment to support continuation of the EUA. But equally important, further vaccine evaluation would be needed to accrue additional data to support licensure of the vaccine as soon as possible and/or to inform labeling. This further vaccine evaluation following issuance of an EUA would include longer-term follow-up for safety, including in larger numbers of vaccine recipients and in populations with lower representation than in clinical trials. Further evaluation would also allow for more precise estimation of vaccine effectiveness in specific populations, and more robust assessment of effectiveness against specific aspects of SARS coronavirus 2 infection or disease, for example, asymptomatic infection. This further evaluation would also characterize the duration of protection, could investigate immune biomarkers that might predict protection, and of course would be ongoing monitoring for signals of enhanced disease. Issuance of an EUA for a COVID-19 vaccine would be contingent upon the ability to conduct further vaccine evaluation, which would occur through a combination of active follow-up of vaccine recipients under the EUA, passive monitoring for clinically significant adverse reactions using established reporting mechanisms, for example, the Vaccine Adverse Events Reporting System, observational studies, including those that leverage healthcare claims databases, and finally, continuation of blinded, placebo-controlled follow-up in ongoing clinical trials for as long as is feasible, and strategies to handle loss of follow-up in those trials. We acknowledge that placebo-controlled blinded follow-up cannot continue indefinitely as more information about a vaccine's safety and effectiveness becomes available. However, FDA does not consider issuance of an EUA for a COVID-19 vaccine to necessitate immediate unblinding of ongoing clinical trials or offering vaccine to all placebo recipients. Of course, trial participants may choose to withdraw from follow-up for any reason, including to receive vaccine made available under EUA. And it may be possible to offer vaccine to placebo recipients in clinical trials in a reasonable time frame that doesn't compromise the integrity of the clinical trial. And these considerations will be discussed further this morning. When an EUA is issued for a COVID-19 vaccine, it will specify conditions of use for which benefit risk has been determined to be favorable based on review of the totality of available data, including those populations to be included or excluded from the EUA, conditions for vaccine distribution and administration, and requirements for safety monitoring and reporting of adverse events. Vaccine made available under an EUA will also include provision of information to vaccine recipients and healthcare providers via prescribing information and fact sheets. These materials will describe that the product remains investigational, will inform about the known and potential benefits and risks, and will also make clear what are the available alternatives and the option to refuse vaccination. Once issued, an EUA may be revised or revoked for a number of reasons. First of all, if circumstances justifying the EUA no longer exist. Second, if criteria for issuance are no longer met. And third, for any other circumstances that arise that warrant changes necessary to protect public health or safety. These other circumstances may be based on new information concerning vaccine safety or effectiveness, vaccine manufacturing or quality, or COVID-19 epidemiology or pathogenesis. The agenda for today's VRBPAC meeting will include, following the conclusion of my talk, first, 3 presentations from the CDC, providing an update on COVID-19 epidemiology, plans for vaccine safety and effectiveness monitoring under an EUA, and operational distribution plans for the vaccine under an EUA. We will then hear about considerations for placebo-controlled trial design if an unlicensed vaccine becomes available. Following lunch, we will have an open public hearing, and then we'll dive into a discussion of the data. First, with a presentation by Pfizer, and then an FDA presentation. At the end of the day, we will have a committee discussion and a vote. We have 2 questions that we would like the committee to consider for discussion. These will not be voting questions. First, Pfizer has proposed a plan for continuation of blinded, placebo-controlled follow-up in ongoing trials if the vaccine were made available under EUA. We would like the committee to discuss Pfizer's plan, including how loss of blinded, placebo-controlled follow-up in ongoing trials should be addressed. Second, we would like the committee to discuss any gaps in plans described today and in the briefing documents for further evaluation of vaccine safety and effectiveness in populations who receive the Pfizer-BioNTech vaccine under an EUA. Following discussion of these items, we will have a single question for the committee to vote on. The question is, based on the totality of scientific evidence available, do the benefits of the Pfizer-BioNTech COVID-19 vaccine outweigh its risks for use in individuals 16 years of age and older? This concludes my presentation. Thank you very much.
Thank you, Dr. Fink, for a very clear presentation. We have a fair amount of time before the next scheduled presentation, which is very good because I think there are some questions of clarification that the committee may have about our guidelines for discussion and for our eventual vote. So committee members, please raise your hands if you would like to ask Dr. Fink some specific questions. We don't want to start our discussion of the points raised by Dr. Fink, but just clarification about the characteristics of an emergency use authorization and the other guidelines. So please raise your hand. I may be having some technical difficulties, so Mike, you may need to recognize the person. Go ahead.
No problem. All right, the first one we have is Dr. Sawyer. Would you go ahead and turn your camera on?
Thanks very much, Dr. Fink. You mentioned that issues related to manufacturing can be one of the reasons to revise or revoke an EUA. What is the monitoring process going forward from this point with regard to manufacturing process?
Thank you. So FDA continues to engage with the vaccine manufacturer concerning manufacturing quality and control issues to ensure that these remain adequate to support use of the vaccine under EUA. I'll invite any of my other FDA colleagues to comment further.
Yeah, this is Marion. Can you hear me, Mr. Chairman?
Yes, we can. We can hear you.
Okay, thank you very much. So no, I just wanted to second what Doran just said. It is true that the monitoring of the CMC, that is the chemistry, manufacturing, and control information, that is currently available and will, and we of course will get additional data from the manufacturing, from the different manufacturing sites. We will review this information and we will work together with the vaccine manufacturer to make sure that the product that is made and generated is of adequate consistency and quality. So this is work that's going to be ongoing over the next month.
Thanks very much.
Okay, Arnold, are you able to see the? Yep, Arnold, are you?
No, I can't. That side of my screen I see, but no, there's no activity there.
Okay, that's all right. So, while you
please, why don't you manage the calling on?
We'll take care of that.
Right.
All right, Dr. Corella?
Thank you. I don't know why my camera is not working, but it says it's on. Doran, just trying to understand the question we're being asked to vote on is what Pfizer has requested, but if I understood your talk correctly, the FDA can limit the actual can place limits on the target populations or the specific indications regarding the EUA. My question is, how do you view the investigational status of the product under an EUA?
Thank you for the question. So a product that is made available under an EUA is an unapproved product. It has not been FDA licensed.
So let me just, if I can just follow up with that. So if you limited the EUA to a specific set of populations, even those populations that have the EUA, placebo-controlled trials would still be possible?
Well, we will have a discussion on those considerations later this morning, but in FDA's view, issuance of an EUA for a COVID-19 vaccine should not preclude the conduct of placebo-controlled trials. In particular, in situations where that vaccine made available under an EUA is available only in limited quantity.
Okay, I think I can see now. Thank you for fixing it. Dr. Lee.
So thank you. One of the questions I have is a little bit more general. I recognize we're considering the Pfizer product for an EUA today. However, future EUA applications, if, for example, there is a vaccine that is approved under a BLA, vaccine number 1, let's just call it that, will that make it more challenging for future products to request an EUA?
Right. So I think what you're getting at is the fourth criterion for issuance of an EUA, which says that there must not be any adequate, approved, and available alternative therapy.
Yes, correct.
So first of all, if a vaccine is made available under an EUA, it is not approved. And so therefore, that would not preclude issuance of an EUA for another vaccine. If a vaccine is approved by FDA, that would also not necessarily preclude issuance of an EUA for another investigational COVID-19 vaccine. For example, if the approved vaccine is available only in limited quantity, then it may not be considered adequate to address the public health emergency. Second of all, if the vaccine that is approved for use only in a limited population, then that vaccine may not be considered adequate to address the needs of the public health emergency for other populations.
Thank you.
Mr. Tubman.
Yes. First of all, thank you and the other members of wonderful job. I think the whole country owes you a gratitude for all.
You speak a little louder, please. We're having problems hearing.
Sorry. Just thanking Dr. Fick and everybody at the FDA for all the work.
Right.
Independently reviewing the data. Thank you. Two questions. One is, you indicated that since the submission came in from Pfizer, you've had a lot of ongoing discussions. And first question is, does that include getting updated data? Because the closing date for data that's in their submission is November 14th, just 26 days ago, which is almost a month, about half of the entire period for the 2 months minimum required for EUA. So have you gotten more recent, requested and gotten more recent data? And the second question is a follow-up to Dr. Perla. The question, you've given us only one question, and that is yes or no to recommending approval for the entire amount, which is population, which Pfizer is requesting, which is the entire 16 and over. But certainly the committee could view this as something they'd like to grant EUA for, but given the balancing of risk and benefit, it may be different for different populations. So we might want to grant it or recommend it for a smaller set than Pfizer's asking for. But your question doesn't seem to allow for that. And I was wondering if you have a backup question that might be used. I know that in the past with the dengue review, there's backup questions. So just want to ask the experience with.
Okay, could I say that your last question, we can bring that up during the discussion later on. So I'm going to excuse Dr. Fink from having to answer that part of the question.
Thank you.
So
to answer your first question, no, we have not received additional data sets beyond the data sets that were submitted to us comprising a cutoff date of November 14th. As you can imagine, there is a tremendous amount of work that goes into preparing a data set for submission. And so it really is infeasible for the sponsor and for FDA to be chasing our tails, trying to get data sets that encompass more and more data as time goes on. That being said, if the sponsor becomes aware of, or if we become aware of any data that would potentially impact our benefit risk assessment, we do have discussions with the sponsor regarding those data.
And we will be able, Mr. Taubman, to ask questions of the sponsor after their presentation later on. So we can revisit this issue. Next, Dr. Perlman.
Yes. So I just had a question about the last part of what you were talking about when you talked about how Pfizer is going to deal with the issue of placebos going off the vaccine trial, and you asked for advice from the committee. Is that part of the EUA vote, or is that a second issue?
So that is not a question for a vote. It's just an item for discussion. We'd like to hear the committee's thoughts on the plan that is being proposed by Pfizer, which you will hear about in greater detail later today.
Dr. McGinnis.
I have a question that's sort of a follow-up from what Mike Carilla originally started. Given that this is considered an investigational product under an EUA, I'm assuming that any manufacturer cannot actually market such an investigational vaccine. Is that correct?
So this is the emergency use authorization does not allow for commercial distribution of the vaccine via the usual marketing that would be available to a licensed vaccine. So I hope that clarifies things. If anyone else from FDA wants to chime in, I'd welcome additional remarks.
So Doran, thank you. I'm happy to get the comments. I'm just trying to understand. So it's not marketed traditionally, but compensation for an investigational vaccine is possible under an EUA, like with a government entity?
So again, this is not my area of expertise.
I understand.
The emergency use authorization may include conditions related to advertising and other similar issues. In terms of transfer of money between vaccine recipients and providers or between the U.S. government and the manufacturer, I can't speak to those.
Dr. Meisner.
Thank you, Dr. Monto. And I would like to thank Dr. Marks, Dr. Fink, and Dr. Gerber and everyone else at the FDA, because the amount of work that you have done is just extraordinary. And the briefing packets that have been circulated are clear and extremely helpful. So thank you very much for your ongoing work. The question I have for you is, it will be advantageous for a number of reasons to have one of the COVID-19 vaccines available under a biologic license application instead of an EUA. And can you, and I realize it's a difficult question, but can you offer any comments about the route or the pathway forward for the FDA to begin to think about when you've reached a point that you would consider a BLA?
Yes. So first of all, we do want any vaccine that is made available under an EUA to continue its testing to allow for its licensure as soon as possible after issuance of the EUA. So this continued evaluation, as I explained in my presentation, I think first and foremost would include some longer-term follow-up of participants enrolled in ongoing studies, as well as safety and effectiveness data coming out of use of the vaccine under the EUA. This additional evaluation would then meet our usual expectations for clinical data to be included in a licensure application. There are some other sources of information that involve manufacturing and facilities and potentially non-clinical studies as well that we would typically require to support a licensure application, but are not absolutely necessary to support issuance of an EUA.
Is it possible to predict or estimate when conditions of safety and efficacy might be satisfied for BLA?
In terms of the time it would take, yeah, I couldn't predict, but I will say that we typically ask for at least 6 months of follow-up in a substantial number of clinical trial participants to constitute a safety database that would support licensure.
Thank you.
And finally, Dr. Fuller.
Overview. My question, did I understand you to say that the FDA in the October 22nd meeting looked at the manufacturing quality of the Pfizer vaccine and that was satisfactory? And if so, how will that be evaluated in the actual distribution of the vaccine? And will we talk about that later?
Right. So to clarify what I said earlier, as part of the EUA submission and in information submitted prior to the EUA, FDA has been conducting an ongoing review of manufacturing quality, consistency, and control. And we have found this information to be adequate to support emergency use authorization of the vaccine. We are not intending to discuss details of the manufacturing process, many of which are proprietary, during today's meeting. But as Dr. Gruber and I discussed in response to a previous question, our review of the manufacturing information is an ongoing process and will continue even after the vaccine is authorized, if that is the decision that we make.
Okay, thank you.
Thank you, Dr. Fink. You kicked us off with a lot of information, which we're going to be coming back to later on this afternoon. I'd like next to call on Dr. Aaron Hall from CDC, who's the co-lead in the epidemiology task force, and he is going to give us an update about the current situation.
All right, thank you very much. Good morning. Can you hear me okay?
Yes, we can.
Wonderful. So good morning. I'm Dr. Aaron Hall, co-lead of the Epidemiology Task Force in CDC's COVID-19 response and chief of the Respiratory Viruses Branch. To help frame subsequent discussions today about COVID-19 vaccines, I would like to provide a brief update on the current epidemiology of COVID-19 in the United States. As of December 8th, over 14.8 million COVID-19 cases and over 280,000 associated deaths have been reported in the United States. The initial peak in early April was driven largely by elevated activity in the New York metro area, followed by a second, larger peak in late July, primarily due to increased activity across much of the southern U.S. Since mid-September, daily counts of new cases have again been on the rise, with even sharper increases since mid-October. Since submission of these slides in advance of this meeting, we have now surpassed 15 million cases and 285,000 deaths nationally, with over 200,000 new cases and over 2,500 new deaths reported yesterday. In addition to reported cases, CDC uses several other systems to track the pandemic, which are compiled in the weekly surveillance summary called COVID View. This weekly report includes the percent positivity of molecular tests for SARS-CoV-2, the virus that causes COVID-19, shown in blue, as well as syndromic surveillance for COVID-19-like illness, or CLI, among ambulatory patients, shown in red. Both of these leading indicators have been increasing since September. Also included in COVID View are weekly hospitalization rates, shown in gray, which are currently at the highest point since the beginning of the pandemic, and also the percent of deaths due to COVID-19, influenza, or pneumonia based on death certificates, shown in green, both of which are lagging indicators that have been increasing since October. One component of the weekly COVID View report is an assessment of COVID-19 hospitalizations through COVID-Net. COVID-Net conducts hospitalization surveillance in 14 states, representing about 10% of the U.S. population. Patients must be a resident of the surveillance area and have a positive SARS-CoV-2 test within 14 days prior to or during hospitalization. Medical chart reviews are conducted by trained surveillance officers, and data are updated weekly on an interactive website. While focused on the more severe end of the illness spectrum, COVID-Net provides active, population-based surveillance, thus overcoming some of the biases with passive surveillance and providing robust data on the epidemiology of COVID-19. Looking at weekly hospitalization rates by age, we see that each of the peaks in April, late July, and currently have been most pronounced among adults aged 65 years and older. Hospitalization rates in children have been considerably lower than those among adults, but have remained stable or increasing since the spring. Note that the last few data points are subject to reporting lag and may increase subsequently. Looking at the cumulative hospitalization rates through late November from COVID-Net and stratifying by age group, we see a strong increasing trend with increasing age. As of November 28th, adults aged 65 years and older had a cumulative rate of 756 per 100,000, which is roughly equivalent to 1 in every 130 people in this age group being hospitalized with COVID-19. This rate is approximately 4.5 times greater than that of adults aged 18 to 49. COVID-19 surveillance has also helped to identify significant racial and ethnic disparities in the rates of COVID-19. Shown here are age-adjusted cumulative hospitalization rates by race and ethnicity, demonstrating rates are 3 to 4 times greater among persons that are Hispanic or Latino, non-Hispanic American Indian or Alaska Native, or non-Hispanic Black or African American, compared with those that are non-Hispanic white. These disparities are likely multifactorial, potentially influenced by differential exposure rates, prevalence of underlying medical conditions, access to care, and other socioeconomic factors. To help further tease apart these issues and identify risk factors for severe COVID-19, CDC and public health partners analyzed the relative rates of in-hospital mortality from COVID-19 using models that adjust for age, sex, race and ethnicity, smoking, and several underlying medical conditions. As shown in the red box, older age was the strongest independent risk factor for in-hospital death, and the risk increased with increasing age. Other characteristics significantly associated with in-hospital mortality include male sex, immunosuppression, renal disease, chronic lung disease, cardiovascular disease, neurologic disorder, and diabetes. Combining data from COVID-19 with population-based data from the Behavioral Risk Factor Surveillance System, or BRFSS, we likewise developed models to assess risk for COVID-19 hospitalization among adults with specific underlying conditions. Again, after adjusting for age, sex, and race and ethnicity, the risk for COVID-19 associated hospitalization was greatest for adults with severe obesity, chronic kidney disease, and diabetes, as shown in the red box. Compared with adults without these conditions, those that have them were 3 to 5 times more likely to be hospitalized for COVID-19. Furthermore, the risk of COVID-19 hospitalization increased with the number of underlying medical conditions, as shown in the red box on this table. While specific risks vary depending on the specific underlying medical condition, adults with 3 or more conditions had 5 times the risk of COVID-19 hospitalization compared to adults with no conditions. Due to increased age, underlying medical conditions, and their congregant living situation, residents of long-term care facilities have been disproportionately impacted by COVID-19. As shown here, residents of these facilities comprise nearly 50% of COVID-19 hospitalizations among adults aged 75 to 84 years, and nearly 2/3 of COVID-19 hospitalizations among adults aged 85 years and older. As such, the Advisory Committee for Immunization Practices, or ACIP, recently recommended long-term care facility residents as a priority group to receive initial doses of COVID-19 vaccines once approved. Similarly, healthcare personnel have been prioritized for vaccination to preserve capacity to care for patients with COVID-19 and other illnesses. Again, based on COVID-19 data, 6% of adults hospitalized with COVID-19 were healthcare personnel, with nursing-related occupations being most frequent. Healthcare personnel hospitalized with COVID-19 had similar prevalence of underlying medical conditions, most notably obesity, as that observed among adults hospitalized with COVID-19, all adults. Likewise, a similar proportion of severe clinical outcomes, including ICU admission, mechanical ventilation, and death, occurred among healthcare personnel as that across all adult COVID-19 hospitalizations. As we prepare for COVID-19 vaccines, it's important to establish baselines to assess their future impact and maintain ongoing assessments of the total burden of SARS-CoV-2 infections in the U.S. To that end, CDC has implemented a nationwide seroprevalence survey to help track the number of people with evidence of previous SARS-CoV-2 infection, including milder infections that do not result in care seeking or testing for acute infection. These involve biweekly testing of approximately 50,000 residual specimens from commercial laboratories for antibodies against SARS-CoV-2. Through the first few rounds of this survey, estimated seroprevalence has ranged from 0.4 to 23% across U.S. jurisdictions. However, as of late September, less than 10% of specimens from most jurisdictions had evidence of previous SARS-CoV-2 infection. In general, the highest seroprevalence was observed among children and adults aged less than 50 years, and lowest among older adults aged 65 and older. Using a different multiplier modeling approach, which uses reported cases and other data sources to then account for underdetection and underreporting, CDC recently released estimates of the total number of hospitalizations, illnesses, and infections with SARS-CoV-2 in the U.S. This analysis estimated that 1 of every 2.5 hospitalized cases and 1 of every 7.1 non-hospitalized cases may have been nationally reported. Applying these multipliers to reported cases through the end of September yielded a national estimate of 2.4 million hospitalizations, 44.8 million illnesses, and 52.9 million total infections. As the figure to the right shows, most estimated hospitalizations occurred among older adults, while most illnesses and infections were among younger adults. So in summary, now, as of December 9th, over 15 million cases and over 285,000 deaths associated with COVID-19 have been reported in the United States. However, based on seroprevalence surveys and models, the total estimated number of infections is likely 2 to 7 times greater than reported cases, though less than 10% of the population in most states had evidence of previous infection through September. Factors associated with increased risk for severe COVID-19 include older age, racial and ethnic minority group membership, and several specific underlying medical conditions. Ongoing surveillance and epidemiologic studies will help inform further development and implementation of candidate vaccines, including assessment of their impacts, safety, and effectiveness. Lastly, even with the promising advent of COVID-19 vaccines, there's continued need for non-pharmaceutical interventions, including mask use, physical distancing, hand hygiene, and environmental disinfection to help bring an end to this devastating pandemic. Finally, I would like to acknowledge the thousands of public health professionals that have worked tirelessly over the last 11 months on the CDC COVID-19 response, including the dedicated staff from the Respiratory Viruses Branch. Thank you.
Of COVID. I think I wasn't on at first. Thank you so much for being clear about the differential impact of COVID-19 in the U.S. population. We have time for relatively few questions. I see Dr. Meisner, do you have your hand raised?
Yes, sir. Thank you very much for that presentation, Dr. Holmes. I would like to ask you about the severity of disease, particularly in older adolescents, that is, individuals who are 16 and 17 years of age, because they have been included in the company's request for an EUA. I I assume it's unlikely that hospitalizations and disease are broken down by age, but is it safe to assume that adolescents who are 16 and 17 years of age are similar in the 5 through 17-year-old age group?
Yeah, thank you for that question. So, of course, as we refine to smaller and smaller age brackets, the numbers get smaller, particularly in children, where overall we see lower rates, particularly of severe disease, of hospitalization. In general, we do see higher rates of hospitalization among children aged 0 to 5 relative to those aged 5 to 17. Of course, this is potentially confounded by differential rates of care seeking. As you might imagine, there are certainly lower thresholds for care seeking for the very youngest and most vulnerable children. However, as we start to look at the more mild end of the illness spectrum and look at rates of detection of SARS-CoV-2 virus using molecular assays, we do see indication of higher rates of infection among older children, among adolescents, particularly then as we move into kind of older teenagers and people in their young 20s. Much of this may have been driven in part by outbreaks in the fall among institutes of higher education, but the broader impacts in children perhaps are not entirely clear until the full resumption of normal activities ensues in the United States, including in-person education across all schools in the United States. So we'll continue to monitor it closely, but thus far the indication is the highest rates of illness, severe illness in young children, but higher rates of infection in older children.
Thank you.
Dr. Rubin.
Thanks, Dr. Hall. I was interested in the multiplier that you described. That applies to the diagnosis of infection. Do you think that applies to deaths as well?
Yeah, so we have used at CDC the same multiplier model previously for tracking influenza and generating in-season estimates of the disease burden. And that same approach has been used previously to estimate deaths in the same manner that I presented today for hospitalizations and milder illness. There are, of course, differential multipliers that would have to be considered for deaths, as the rates of underreporting of death are different than those for milder infection. In general, we have better capture of deaths. We have lower rates of underreporting and under-ascertainment for deaths. But as folks are aware, the dynamics of the pandemic itself have greatly changed those multipliers. And so there's also a considerable time component that needs to be factored in when using these multiplier models. So the underreporting of deaths, for example, has changed over time, and the attribution of deaths to COVID-19 has changed over time, which complicates interpretation. But we do have several efforts underway using these models and other approaches to generate estimates of death. And we do feel, as with hospitalizations and illnesses, that the reported number of deaths is likely an underestimate of the true number of deaths.
Thank you.
Thank you. Dr. Gantz, we're going to go over a little bit, but please keep your questions short.
Thank you very much. Thank you for that, and I agree, heroic effort to all of the people who are working on this. I had two quick questions. The data concerning the immunocompromised or immunosuppressed individuals is not really granular enough to make it something that we can use in terms of how we're thinking about high-risk populations. There are registries looking at this, but it would be nice if there were some national information that you could provide to us, as well as I didn't see any information from pregnant women, which is a population which we're all concerned about, and we've definitely seen postnatal transmission to very young infants who end up being hospitalized. If you would just take those up.
Yeah, thank you for those comments. Absolutely. There are numerous surveillance efforts currently underway to assess the impacts of COVID-19 in pregnant women. In the interest of time, unfortunately today I didn't have a chance to present those, but through COVIDNet as described today, as well as another surveillance system called SETNet, which was established during the Zika epidemic. We have been very closely monitoring the impacts of COVID-19 in both pregnant women and subsequently following up with their infants. Thankfully, the rates overall have been relatively low thus far, but as we continue to accumulate a critical mass of data in this demographic, we indeed do hope to have more specific estimates of the risks that are posed to pregnant women and their infants. The early indication is that there may be a higher risk of preterm delivery among pregnant women infected with COVID-19 relative to women without COVID-19. But there's ongoing efforts to assess those and other potential pregnancy-related risks and fetal outcomes.
Thank you very much, Dr. Hall. And it's now time for our morning break. We've eaten into the time a little bit, but I do want to start again at 10:30 Eastern, so we can hear from Dr. Messonnier from CDC. See you later.
All right, welcome back from break. We'll now enter our second half of our next group of speakers. Arnold?
We're next going to hear from, again, from the Centers for Disease Control. We're going to have a few presentations and questions afterwards. First, we're going to hear a presentation from Dr. Nancy Messonnier, the Director of the Center for Immunization and Respiratory Diseases, on vaccine safety and effectiveness monitoring. And then from Anita Patel, the Deputy of the Vaccine Task Force on Operational Distribution Plans. Dr. Messonnier.
Thank you, Dr. Monto, and thank you for the opportunity to speak today on this really auspicious day. Despite the completely appropriate size and scope of the clinical trials, it's always important to continue to monitor vaccines post-licensure, post-authorization, and that is certainly especially true with COVID vaccines. So I'm going to talk about the U.S. government plans for monitoring of safety and effectiveness. First, safety. The rapid implementation of safety monitoring under an EUA requires a whole of U.S. government approach with an initial focus on the early populations targeted for vaccination. Voluntary active surveillance of adverse events focused on healthcare worker vaccination and rapid follow-up of reported serious adverse events. As the program continues and more vaccine is given, active surveillance systems will provide increasingly useful information on safety in different populations. Close collaboration of safety experts across the USG will facilitate data sharing and rapid recognition of responses to safety signals. Now, there is a quite extensive plan for safety monitoring, but I'm just going to summarize a few high points. On day one of the COVID-19 vaccine programs, systems will be in place to monitor the safety of vaccine recipients. These include the traditional systems that we use, VAERS, which is a National Spontaneous Reporting System monitored and implemented by CDC and FDA, but also similar programs at DoD and the VA, as well as the National Healthcare Safety Network, or NHSN, which is an acute care and long-term care facility monitoring system. CDC CSIS system is stood up that provides individual case consultation. But in addition, to really enhance this period, CDC has stood up a new system called vSafe. It's an active surveillance tool that uses text messaging and web-based surveys to monitor vaccine recipients for adverse events. Recipients complete brief surveys up to really intermittent surveys to 12 months following vaccination. But then as part of the program, recipients that report a significant health event that impacts their daily activities or leads them to seek medical attention will be called for more information and a report will be generated. This is an opt-in system, and so we're going to have a heavily enhanced engagement around implementation of the program. We really need people to sign on to this system to give us the best data possible. Oops. Large linked database monitoring systems will provide safety data when vaccine becomes more widely available in the priority groups and the general public. This includes, again, existing systems like VSD, the FDA's extensive system, and systems from DoD and VA. We're enhancing this by Genesis, which is a new collaboration with NIH and Brown University to monitor safety in long-term care facility residents. This system operates nationally and has near real-time data for 250 facilities. To look at this another way, this is a table that showcases the systems that we'll be using to monitor safety in the populations that, based on ACIP recommendations, will be vaccinated first, specifically healthcare workers and long-term care facility residents. So you'll see that early on, we have a series of systems to monitor healthcare workers and long-term care facility residents through VAERS. VAERS will be enhanced in this time period especially around long-term care facility residents, to ensure that we're getting those reports. And then Be Safe, which will specifically target healthcare workers. And then you'll see all the systems that are going to be in place for later monitoring in all of those populations. I think the team has done a really good job of thinking through what systems we have, how to enhance those systems, and looking for any weaknesses and attempting to fill them in. And, you know, I'm giving it short shrift by going through it so quickly, but hopefully you'll understand how robust these systems are. In addition, the ACIP COVID vaccine safety technical subgroup was established to provide expert consultation on COVID-19 vaccine safety issues. This group, which we call VAST, was built off lessons learned during H1N1 safety monitoring. The terms of reference and composition have been finalized, and VAST is ready to begin reviewing data once implementation commences. The group is co-chaired by a member of ACIP and a member of the National Vaccine Advisory Committee, or NVAC. There are 10 independent expert consultants that make up the group, as well as ex officio members from NIH, FDA, OIDP, CMS, HRSA, and IHS, and we also have representatives from the VA and the DoD. Now to briefly summarize our efforts around post-authorization vaccine effectiveness. Post-authorization or post-licensure VE estimates are needed to address important evidence gaps from phase 3 clinical trials, particularly for secondary endpoints that may not be adequately addressed. For example, the trials may be limited in their ability to address a VE against secondary endpoints like infection transmission or in subpopulations, and we won't have great data early on the duration of protection. We also believe it's important to evaluate the real-world performance of vaccines, as protection may differ from efficacy under trial conditions. So, this table attempts to lay out the VE policy priorities that were determined based on consultants with policy experts and internal and external experts. The most immediate priority is to determine whether the vaccine protects against symptomatic disease as expected based on the phase 3 trial data, but understanding that implementation for these vaccines might be slightly complicated by folks who don't exactly get the second dose exactly on schedule and by the necessity to keep the vaccine in cold chain. So that's going to be the focus, but then there are a variety of subsequent and late stage studies that are being planned or implemented. A number of strategies are used to plan for the assessment of VE, but to facilitate a rapid launch, we've leveraged existing platforms, and for early stage vaccination, we focus on populations likely to be vaccinated first. We're harmonizing and coordinating across the U.S. government on a variety of platforms, for example, using common case definitions, data elements, and methods to improve comparability of results. And we've been working to combine similar platforms to improve geographic representation, increase statistical power, and generate more timely and robust VE estimates. And we do have a diversity of methods as we understand that all observation methods have limitations. Here's a summary of the studies that are being planned. And again, I don't have time to go through all of them, but I would just put you to the top row. You know, for the most immediate question of whether vaccines work as expected, we're planning a prospective study among healthcare workers. There is no large existing database enriched for this group, and so we're doing a test negative design case control study among healthcare workers, which should help us relatively quickly assess this outcome. So, in summary, planned VE studies will provide a robust assessment of COVID-19 vaccine performance in real-world settings. We believe we will have early VE estimates in groups prioritized for vaccination, VE against key outcomes, and in important subgroups. And data from these studies will address critical gaps and help guide future use of vaccine. Systems are in place to monitor the safety of COVID-19 vaccination in healthcare workers and long-term care facility residents. VSafe, VAERS, and HSN will detect adverse event signals for further follow-up and evaluation. CMS, Genesis, and other claims-based and EHR systems will be used for both signal detection and evaluation. Vaccine effectiveness in healthcare workers is the immediate priority and will address the question of does the vaccine work as expected. We expect those studies to start immediately. And vaccine effectiveness evaluations in older adults, including those in long-term care facilities, are planned and include both test negative design and cohort evaluations. I hope that does justice to all the systems that we're planning and that are in place. Understand that vaccine safety and effective monitoring is a top priority for the U.S. government, and we're committed to ensuring that all these systems are in place and ready to go as soon as the vaccine program is implemented. Thank you.
Thank you, Dr. Messonnier. We're going to go directly on to Dr. Patel's presentation, and then we'll have questions afterwards, so we have plenty of time. Dr. Patel.
Great, thank you so much. So thank you for having me today. And we'll be walking through today at a high level, what the USG's distribution strategy is for COVID vaccines, specifically focusing around the Pfizer vaccine product. The USG plan for COVID vaccine distribution will adjust as volume of vaccine doses increases. Early on, we do anticipate that we'll have a constrained supply where vaccine administration is really targeted towards select population groups, and distribution will facilitate that. As sufficient supply becomes available and the populations that we're trying to reach expand, our distribution network will also further expand. The schematic here outlines what our concept is for distribution at a very high level. All vaccines will be ordered through CDC's COVID vaccine ordering systems. For the Pfizer vaccine, we will be providing those vaccine orders directly to the manufacturer for distribution to administration sites. We've been working very closely with our state and local partners to make sure that they have plans in place to identify providers that are able to receive, store, and use the vaccine for the specific populations that they're trying to reach. These administration sites will be receiving the Pfizer vaccine product, and they should expand as well over time as the targeted populations we're trying to reach expand. In addition to the hospitals, the vaccination clinics that have been set up to support early distribution and use of Pfizer vaccine, we have also set up a program called the Long-Term Care Facility Pharmacy Program. This federal program is a partnership that allows for jurisdictions to be able to opt into the program, allowing for on-site vaccination support to occur to long-term care facilities. Our pharmacy partners would be providing the on-site vaccination support for those facilities that actually sign up for the program. Also like to draw your attention to the Operation Warp Speed Coordination Cell at the bottom of the slide. The idea is for us to be able to have a clear understanding of what is happening in terms of supply of vaccine as well as administration and uptake information throughout the vaccination program. Focusing a little bit more on the long-term care facility plan, in order for us to be able to reach skilled nursing facilities, we have been working to bring skilled nursing facilities on board to the program and opt in. Once a state determines that they're ready to expand vaccination to reach staff and residents within skilled nursing facilities and other assisted living facilities, the program would be turned on. Right now, we do have a very high percentage of the skilled nursing facilities within the United States that have opted in to receive services from this federal program. The vaccine characteristics of the Pfizer program are very different and unique compared to other vaccines that we've seen before, and they do impact distribution, site storage, as well as administration requirements. The details that's depicted on the slide are very specific to the Pfizer program and outline some of the challenging requirements of the vaccine that have to be implemented in terms of vaccine distribution and storage sites. The additional planning that is needed for distribution, storage, handle, and use has been underway for the last few months.
based on the information that we know for the Pfizer product. There is a required dedicated cold chain given that the product has to be managed at ultra-low temperature requirements. In addition, the timing of the vaccine ancillary supplies, which include the diluent as well as dry ice replenishment, has also been a challenge. We have built the distribution system in a way that all three of these elements will arrive within a 36-hour period of time. So once the vaccine is there, these additional pieces will also be delivered to the vaccination sites. Another major challenge that we've had with the product is the actual minimal order requirement. 975 doses is the smallest volume of product that can be ordered for the Pfizer vaccine. This has posed challenges, especially in rural areas of the country where that volume of product is more difficult to manage. And it's also been a planning factor that at the local level has been used to determine where exactly the product should be received. There's also a new element of the thermal shipping container that has been introduced with this product. The container allows for the movement and shipment of the vaccine from one site to another and allows it to be managed in the ultra-low temp conditions. There is training required with using this thermal shipping container and staff being able to be trained on the monitoring, the dry ice replenishment requirements, as well as understanding how to manage the product over time. How many times to open the container and the details have also been part of the training and what we're getting vaccination sites ready for. There's also complexities with mixing and using the product. It is a little bit different than other vaccines that we've seen as far as the diluent being required, how the product is drawn up, and also how quickly the product needs to be used. Once diluted, we do have a 6-hour period of time in which the vaccine needs to be administered in, and that has also been factored in and built into the planning at the administration site level. Lastly, we do have inventory planning that is underway to ensure that there is sufficient supply available for administration. This includes ensuring that administration sites have actual clinic planning times, that they're able to have enough people come to manage the 975 doses for each of the trays, and the planning for the actual clinics, as well as the planning for the second dose. So second doses are required, the timing, 21 days, and any cushion that we have beginning or ending from the 19 to 23 days that we saw in the clinical trials. What exactly that looks like and how that gets operationalized and implemented is also part of the planning factors. Second dose reminders are part of ensuring that people come back for that second dose, and additional strategies are also underway. So with that, I'd like to pause and turn it back over to the moderator.
Thank you, Dr. Patel. We're going to have time for just a couple of questions at this point. Dr. Levy.
Hello. I have a brief question for Dr. Messonnier. First of all, thank you so much for your presentation. It was very helpful. You needed to move very quickly through some very important information about the safety surveillance systems that are in place to monitor the progress of coronavirus vaccine. I did have a question. You described a number of interlocking national systems that would mirror vaccine safety. I wanted to know if there's any consideration or plans for also integrating these systems with international efforts at monitoring coronavirus vaccine safety. For example, World Health Organization, WHO. There's also an international network of special immunization services and others. So that's my first question. My second question has to do with vaccine effectiveness. You mentioned some key subgroups that you'll be monitoring vaccine effectiveness in. Those all made good sense. It went by quickly, but I didn't see teens or maybe even eventually younger children listed among those special populations, but I would imagine they would be of interest as well, and I'd value your comments on that. Thank you.
Thank you for easy questions. The answer to both questions is yes. There's actually a WHO coordinated group looking at sharing data for both safety and effectiveness and coordinating across the globe on that. In addition, we are working directly with collaborators in the UK and in Canada to make sure that we're sharing information as quickly as possible. And we're already gearing up to help a number of countries that are for immunization campaigns. The answer to the second is definitely yes. I didn't put it on the slide because authorization for those groups was not really up for discussion today, but certainly it will be really important to be able to monitor both safety and effectiveness in an enhanced way in those groups. Yeah, period.
And the final question from Dr. Kurilla. After that, we'll have to move on.
Thank you. This is for Dr. Messonnier. You described a very comprehensive assessment, aggregation, and collation of a lot of safety signals, and I'm wondering, the intention for that group, is that to provide ongoing advice to the people who would be distributing the vaccine, potentially administering it, public health officials, or is that primarily to be focused towards the FDA in terms of refinement of the EUA itself? What's the real focus of all of that safety assessment? Where's it going?
I mean, I think it can be for multiple uses. However, our standing up of an ACIP work group is to be a part of our ACIP recommendations, that is the recommendations for use of the vaccine. CDC and FDA and all the USG partners coordinate, collaborate, work together in the safety landscape every day on all our routine vaccines. And as you look at our systems, you'll see how intertwined they are. So any information, for example, that we get will be shared quickly with the FDA, as well as putting these systems in place so that our advisory committee can review it. I also might say that CDC and FDA are also discussing a contract with NASEM, the National Academy, so that we also have them to provide expert independent review of vaccine safety issues that may arise in the COVID-19 vaccination program. So in addition to everything we've set up, we're also setting up that collaboration contract with NASEM so that we also have them ready if we need to help adjudicate a safety signal. Thank you.
Thank you.
Well, thank you to both of our CDC.
Thanks, Sean. Thank you for me to this advisory committee today on one of its most important decisions. And the title of this slide is exactly what I'm speaking to. There we go. So what are these considerations? Well, there are two, and they're intertwined. First are the ethical considerations, which concern the questions about what is the right thing to do with the placebo group in this case. The second class of considerations are epistemic, concerning what we know or what we believe, which is determined by the strength and totality of the evidence. That's what's generated by the designs we are already using and the ones we'll use going forward, which might be the most consequential thing that we discuss today. Now, I will be speaking about ethical issues, but I am not a card-carrying or PhD-carrying bioethicist. But I've enjoyed working with many, and I want to acknowledge, before I talk, the contributions several have made to my remarks today, although the views and ethical lapses are my own. They are Professors Nancy Cass and Ruth Baden from Johns Hopkins and David Magnus from Stanford. Now, there are a lot of questions embedded in what should happen with the designs and particularly the control groups in studies going forward. This slide outlines a lot of sub-questions, but I think the title of the presentation pretty much captures it. If an EUA for this and other vaccines are granted, what do we do with the placebo control groups in this trial and potentially in ongoing and in upcoming trials for the next vaccine? Now, I want to start with a set of ethical preliminaries. I'll start with something that seems very basic, but I can't tell you how many committees I've been a part of and that I've watched whose conversations have founded on this simple issue. We're going to be dealing with ethical dilemmas here, but it's worth thinking about what an ethical dilemma is. So an ethical dilemma is not a choice between right and wrong. That's what we write to advice columnists for. An ethical dilemma is a choice between two actions that are both ethically justifiable under different moral frameworks or principles. In that sense, it's a competition between two rights, two right actions, which of course is exactly what makes it a dilemma. We face this in every clinical trial, particularly on data monitoring committees, the tension between actions that are good for an individual, which in the current setting might mean giving them a vaccine immediately, and what's best for society, which might mean holding off on that vaccine so placebo-controlled randomized trial can continue. Resolution of ethical dilemmas always requires compromise that balances the competing dictates and minimizes physical harms or ethical wrongs which are caused by unfairness, violations of autonomy, or actual personal or societal harm. Now, to continue, our ability to conduct clinical trials, which is to experiment on each other, is something that society gives us permission to do. We don't need that permission to do chemistry or biology experiments. And because the procedures and oversight for RCTs are so structured and standardized, it's easy to forget how fragile that permission to conduct RCTs is. But anybody who's worked as an institution whose research enterprise has been completely shut down because of a single ethical lapse knows that this trust can never be taken for granted. And it's continually earned and re-earned through fair and transparent processes to determine what's acceptable. And that's what we're doing today and why it's so important that we're doing it in public. Now, final preliminaries, which is ethical acceptability depends often on context or background conditions. Offering a placebo subject the vaccine can be judged quite differently depending on whether the evidence, we have about efficacy and safety, and whether or not that participant can actually get the vaccine outside of the trial. And both of those are part of the context today. Now, epistemic preliminaries. I'll start by saying something that shouldn't be too controversial, which is that RCTs, or randomized controlled trials, provide the most reliable knowledge about the relative efficacy and safety of interventions, at least as permitted by the sample size and follow-up time. Obviously, we can't detect safety issues that come up after the RCT closes. A second principle is that reliable knowledge can come from non-randomized designs, and Dr. Messonnier just introduced us to some of those. This includes exactly the kinds of things she described: vaccine safety surveillance, all of epidemiology, quasi-randomized designs, natural experiments, and understanding of biologic mechanisms. These can play a critical role. And finally, there are many things to learn about these vaccines. We've talked about some already. Some of them are best learned through an RCT, but not all of them. And there are many things we can learn from these other designs that do not require RCTs. Now, finally, in this stage setting, there should be no bright lines. That is, bright lines are almost always unjustified, and they are the enemy of ethical and epistemic compromise. In ethics, this means we can't allow one moral framework or principle to trump another. We can argue about how one principle takes precedence over another, but it's not helpful to hear assertions like, it's unethical to deny an effective vaccine to a placebo recipient. Asserting that anything is unethical effectively shuts down the debate, and it also demonizes people on the other side. And I found it extremely healthy for committees not to use that word at all. There's a corollary in epistemology where we can have reasonable discussions about what we can learn and what we can't learn from randomized and non-randomized studies, but saying things like you can't learn anything from a study without randomization is both obviously not true and inimical to a balanced solution. Now, what do RCT participants know? Let's get into the issue of ethical obligations. All consent forms and consent processes are required to tell the patient or the participant in the study that the purpose is to produce scientific knowledge that benefits society, not necessarily them directly. They're also told that they trust that they won't be exposed to known serious harms not listed in the consent, and that they can withdraw at any time for any reason, and that they'll be given information arising during the trial that's highly relevant to their decision to continue. This is exactly what is in the Pfizer consent. In quite plain language, I've put it up here. I won't take the time to read through it, but it's quite explicit, and we have no reason to believe that the study was misrepresented in any way. Here in the highlighted area, you can see you're free to stop participating at any time, and the study team will tell you in a timely manner if new information is learned that would change your mind about continuing in that study, and that certainly would be an EUA. Now let's talk about, I'm sorry, skip the slide there, whether there's special obligations to placebo recipients. Obviously they have exactly the same, we have the same obligation to them that we have overall to the trial. They're free to withdraw, and because of that, the trial structure investigators shouldn't actively deny them vaccine in any way if it became otherwise available to them through a social prioritization and local circumstances. And this is consistent with the FDA position stated earlier. We'll come back to this. I would also argue that the investigators are not obligated to provide immediate vaccination in the trial before their turn of the participant is called outside the trial. First, this is not what they signed up for, and you know, there are many prioritization criteria out there by very, very good groups, and I haven't seen one that includes trial participation as a basis for jumping the queue. That is not considered a reason to get it before somebody else who might deserve it or need it because of higher risk or other considerations. But it should be reasonable when a trial participant becomes eligible through societal priorities and local ability, that they are then can be provided vaccine in the trial context. So in a sense, they jump to the front of their own line. And that also binds them to the trial. It's a form of reciprocity, and it facilitates further follow-up. Now, what are some issues related to the COVID-19 context that will have relevance to these decisions? The first is the most obvious. We're in a pandemic. It's a public health emergency, which magnifies potential consequences, both positive and negative, and knowledge or ignorance of, and the consequences of knowledge or ignorance, and of action or inaction. I would say that some rules that apply in peacetime might have to be modified in war, and we're in a bit of a war right now. But I want to focus specifically on the health risks to individuals. Given the recent numbers, not yesterday's, which are particularly terrible with over 3,000 reported dying, but those in the last few weeks, a randomly chosen U.S. citizen has an average risk of dying from COVID in the next 6 months, that is by the end of May, of roughly 1 in 1,000, and the risk of hospitalization of roughly about 1 in 200. These numbers obviously vary widely by individual, and they don't include the harms from non-fatal COVID, but they're highly ethically relevant, and let's start to talk about that. So if the maximum, if the risk of death in the U.S. in the next 6 months is around 1 in 1,000, that means the maximum benefit of the vaccine for an individual, at least with respect to mortality, is also 1 in 1,000. So, or 0.1%. So imagine if we had a drug that purportedly raised cancer survival from 40% to 40.1%. If there was more to learn, and we do have that here, that's a setting where we would typically allow many placebo-controlled trials. We see this in meta-analyses all the time with multiple trials, placebo-controlled trials of agents with therapeutic benefit orders of magnitude larger than this. So while this risk obviously has huge population impact, in terms of trial ethics, it's not the kind of risk that incurs a great ethical debt to the placebo group or a prohibition on further placebo-controlled trials. Also, unlike a therapeutic drug for patients with a disease who can't change their own prognosis, many, but not all individuals can take actions that reduce their own risk of death from COVID, like masks and social distancing. So as long as there's still important things to learn about the vaccine, placebo-controlled trials should not be regarded as unethical. However, a big however, they might be infeasible. And that is a big issue because people may not be willing to either remain in the study or to enroll. But it's very important to not conflate the feasibility with the ethics. Now, in terms of the vaccine context, what we know, I'm actually not going to spend my time on this. You're going to hear, you've already heard a lot about the Pfizer vaccine. The only one I want to highlight is that if a participant wants to unblind themselves in the trial, they can, if they have the resources and knowledge about which antibody test, commercial antibody tests would unblind them. Some of them are to the spike protein, which would unblind them. Others are to the nuclear capsid, which might not. Now, in terms of the vaccine context or background conditions, what we don't know, well, this has actually already been covered. There's a whole raft of things that we don't know yet and that we want to learn, both from continued RCTs and also from observational studies. Now, I want to outline a possible adverse scenario. If placebo participants who want vaccine when otherwise available and eligible are not given access within the trial in the name of scientific rigor, there may be many who respond to appeals by investigators not to seek vaccine, even if available to them. And to them, this argument doesn't apply. And we've seen that willingness to hang in studies with placebo controls in many other trials of even greater personal consequence. So it's not at all unlikely that there would be a sizable number of people who wouldn't do that on appeal. But some participants at higher risk will want the vaccine as soon as it becomes available to them. And if they can't get it through trial mechanisms, many will lose trust in the investigator and the trial itself, may leave the trial without further follow-up. They may seek vaccine outside the trial. And as more vaccines come on the market, perhaps they'll get another one that's available regionally with very uncertain effect. This will get a lot of publicity, meaning that recruitment to future vaccine trials will be that much more difficult because of a collective loss of trust. So some might appeal to the experimental status of the vaccine under the EUA as justification, as ethical justification for maintaining a placebo control. I understand why this has salience within the FDA, but I suspect that this distinction will not get much traction among future or current or future trial participants when literally millions of people are being urged to get vaccinated as quickly as possible. The point is that once the vaccine is widely available, or any vaccine, widely available and encouraged, the ability to maintain double-blinded placebo-controlled groups voluntarily for more than a nominal period, by which I might mean perhaps a few months, will likely not be under the control of the sponsor of the FDA. And some efforts to do so could undermine future trials if people don't trust that their interests will be watched out for. Now, I want to mention an important countervailing view, which is that many people out there, as we well know, are very wary about an expedited approval process and refuse a vaccine that they believe to be under-tested. They want to see those trials going to their planned end, but unless these people can be found and are willing to enroll in placebo-controlled trials, the argument about the feasibility of the trial still holds. Now, what's the Pfizer proposal for continuation? Now, it implicitly recognizes this reality, and it's consistent with everything I've discussed so far, except in one important regard. First, they propose urging as many people in the trial to stay as long as possible, which I think is the right thing to do. For placebo recipients who become eligible according to local or national recommendations, that's an important condition, and who ask for the vaccine, they propose unblinding them and vaccinating. For those who stay in the trial blinded, it's proposed that everyone be vaccinated after 6 months and followed for an additional 18 months to the trial's planned end. This is coupled with a robust observational pharmacovigilance plan for long-term safety monitoring that augments what the FDA and CDC are doing. While not unreasonable, I think that we have to remember that this design will not only unblind placebo recipients, will also unblind vaccinated participants who make the same request, because of course they won't know. And once unblinded, risk behavior can change. Many of the benefits of randomization within the trial are lost. So is there an alternative? And I would say there is and should be considered. There are two alternative designs that achieve the same ethical goals without compromising without compromising on the epistemic ones. That is, we can give people vaccine who are eligible and who want it, but maintain the blinding, thereby preserving trust that the investigators and regulators care both about the science and the participants. And let's talk about those designs. One is called deferred immunization. It's been presented in a variety of settings and gotten coverage. Also a blinded crossover design. And the second are active control designs, which are really not the issue for today, but I'll discuss them briefly. The first of these was developed by a biostatistician at the NIAID, the NIH, named Dean Follman. It involves blinded crossover designs. And the idea here is that the placebo, when either a placebo or vaccine recipient becomes eligible for a vaccine or requests it, exactly the situation that the Pfizer proposal describes, that the placebo recipient get the vaccine and the vaccine recipient get the placebo without being told what they were originally. So the blind is maintained. And then they are followed forward. So they stay within the trial confines and to some extent also protected by the randomization. As I mentioned, the second design is active control. At some point in the evolution of vaccine testing, we may no longer be able to conduct placebo-controlled trials of any type in settings where vaccines are available. Although it's not a question for today, at that time, when there are a lot out there, a comparison vaccine with reasonably well-established properties may have to be used in one arm in those areas where they're available. So we're not at that stage yet, but we may be within the next year in some countries. Now, in countries or regions where vaccines have seen poor distribution, it may still be possible to conduct placebo randomized trials, but that has ethical complications of its own, and I'm not going to get into that here. I want to talk just a little bit more about the deferred randomization scheme, and you see that in this picture. The idea is that we start with the vaccine and placebo arm as we already have, and then at the point when people are eligible and ask for the vaccine, you have to have both conditions. That's the point where a blinded crossover occurs. Because every participant is ultimately assigned to the vaccine arm at some point, this design actually, in some situations, in very plausible situations, can have greater or equal power to detect the duration, the sustenance of immunity, more than a standard 50-50 RCT. And you can sort of see that in the figure where you see the contrast between the vaccine efficacy in the width of the bar in the delayed vaccine arm compared to the waning efficacy in the original arm. And this increases over time, and these can be compared statistically. So you can see how this can work for vaccine efficacy. But of course, it decreases power to detect some other things like long-term safety issues and some other important parameters, but it doesn't lose them completely, as completely as designs where subjects are unblinded and effective randomization is lost. So if the standard 50-50 RCT is not reasonable or feasible, which I believe it very quickly won't be, this design is far better than any that would break the blind and break the randomization. We can, of course, also reduce some of the ethical tension in any in placebo-controlled trials by increasing the allocation ratio to the vaccine arm, but that may not be enough to solve the enrollment problem. The next slide shows another picture of this deferred vaccination schema, and it shows that it's possible. You see the crossover on the right. On the left-hand side, we have the trials as they've designed so far. On the right-hand side, you see what they look like after the crossover, which is that everybody's been immunized. But it's possible statistically to impute an unvaccinated control group to help estimate secular trends. So to finish up, I want to summarize that these alternative designs require compromises, the kind of compromises I talked about before, on both the ethical side and the epistemic side, that is the evidence. Ethically, placebo recipients don't get a vaccine as quickly as some might want, and this will become more of an issue going forward as new vaccines need to be tested with multiple ones already available through EUAs. But of course, those subjects are not being forced to enroll. But the ones who do enroll can expect to get immunized at a certain number of months going forward. As noted, placebo-controlled trials may continue to be possible in areas or populations for whom vaccines are not available, but that is complicated for other reasons. Finally, on the epistemic side, with this crossover designs, we don't learn as reliably or as quickly about long-term safety and infectiousness and other factors, although we still might learn as much or more about durability when compared with a trial that continues with the placebo arm. But as I said, that trial may not be doable. We might not have the perfect design going forward, and continued blinded observation of the deferred vaccination group can still be tremendously informative and potentially more informative than one with broken randomization, particularly in combination with observational studies of the population that we've heard about today. By reducing the ethical tension within the trial and by retaining the trust in trial testing system, it provides a way further for future studies of other vaccines. So none of these are the last words on this very difficult and complicated subject, but for today, these will be my last words. So I hope this will be helpful to you, and I welcome your questions and comments. Thank you.
Thank you, Dr. Goodman. You've given us a lot to think about and to discuss. I just wanted to start by raising one question. With the blinded crossover design, when would that take place? When an individual becomes reaches the priority listing for their group, or would that be at the time of issuance of the EUA?
So this is a subject of tremendous controversy and discussion. For the reasons I've already outlined, I don't think it should be, and this is my opinion, at the moment of the EUA. I think because otherwise they are jumping the queue, and I discussed that earlier. I believe it should be when for those subjects, they come up on the social they come up as they would come up in terms of priorities for the vaccine, and it's available to them in their area. That is, they can get it outside the trial. As soon as they can get it outside the trial, then this would kick in, because we want to keep them in the trial. If it's given at another time, we're basically giving them priority over other populations that have been deemed to be at greater risk or greater need. And the price I think we have to pay by compromising the trial, I believe would be too high, and we don't have, again, I made this argument earlier, I don't think we have that special obligation to give them the vaccine immediately. Those are my views.
Okay, thank you. I think we'll defer questions about how feasible this is to the sponsor after the sponsor's presentation. Next,
Yep.
Dr. Levy. And please unmute and show yourself if you'd like.
Sorry, that was an error. I don't have a question.
Okay, Dr. Meisner.
Go ahead, Dr. Meisner.
I guess I answered all questions.
All right. Should we go to the next one?
Yeah, Dr. Chatterjee.
Yes, thank you, Dr. Goodman, for your presentation. It was certainly thought-provoking. I have a question with regard to the current study participants. By reviewing the documents, the briefing documents, it appears that there's a difference between participants who received the active vaccine versus placebo recipients in terms of adverse events that they experienced. And so there's a potential that people who are participating in the trial can actually, guess which arm
Yes.
they were randomized to.
Absolutely, yes.
Yeah, so is there then a possibility that those who believe that they were placebo recipients might wish to withdraw in greater numbers, and that would then compromise the study as well?
Again, it absolutely could. So even going forward, it could, and that would be an issue for the statisticians and for the FDA. The point of this particular proposal is this would give them an avenue not to withdraw. That is, stay within the trial under these conditions. Now, if they didn't want to wait for their allocation to become available and their eligibility locally, withdrawing wouldn't do them anything. It wouldn't get them the vaccine any earlier. So there would be no motivation to withdraw. And also, I do think that the majority of people who signed up for these trials did so in good faith and with altruistic motives. I do believe the vast majority want their contribution to produce a valid scientific result. So I don't think self-interest completely in any way dominates. And again, yes, I do believe that there's a fair amount of unblinding just due to reactogenicity of the vaccine, and obviously non-reactogenicity of the placebo. But this, again, at the time when they might be able to get the vaccine outside the trial, this would give them a way to still contribute to the science of the trial without leaving. I don't know if that answered your question.
Yeah, no, that did, and I was thinking about the people who would meet the priority criteria. Obviously, they must be the ones who would want to withdraw if they thought they were in the placebo arm.
Correct, and I do believe that's what I said. I do believe that could happen.
We've got a lot of questions. I think and what we're going to do is I'm going to ask all the speakers to please be available during our discussion, because we may have additional questions that come on up at that point. I'd like to move on to Dr. Kurilla, please.
Thank you, Arnold. A great presentation. So the blinded crossover design looks quite feasible. However, the caveat is that it does take longer to get to the answer. Did I hear that correctly? That's number one. But would we also be facing a first mover effect where this would basically dictate future trial design short of any vaccine having a full BLA going forward? So everyone else will have to consider this option as a de facto approach.
Yeah, that's a really good question. That's why I emphasized that. I'm sorry, I see you talking, but I don't hear you. I'll just answer what I heard. I can't judge exactly what will happen in the future because fact patterns are going to change. So I hesitate to predict too much. It is possible that this might represent, as we go forward, a default design that reduces the ethical tension of these trials as vaccines, as I mentioned, become available in the area of potential enrollees in the trials. And what we'd then be asking them to do is not completely deny themselves a vaccine, which they could just go out and get if they themselves are eligible, but just defer it for a few months. Might be 2 months, 3 months, 4 months, and that might be less of an ask. I can't say whether it will become the default. Again, it's going to be a while before enough vaccines of any type, no less just this one, are rolled out, even in the U.S. Remember also that these trials are going on all over the world. So the U.S. Places where they will not always be occurring in places where vaccines are otherwise available for quite a while. So it is possible we might evolve. I mean, I do see that there's going to be a staging where there's going to be a point or places where we cannot any longer do the placebo-controlled randomized trial. We might move to this, and then ultimately, as I said, and it might be a year or two from now, have to move to active controls. That's the evolution I see. Whether you're committing to this design going forward, harder to say. That's very complicated. And again, it's going to depend a lot on vaccine distribution and what we learn about these vaccines. If there start to be safety signals that we're not seeing right now, that could change the dynamic dramatically.
Thank you.
I'm just going to allow one more question because we have to start at noon, which is difficult to. So Dr. will be the last question, and we're discussion as part of we'll really want to pick where we've left off on some of these issues at that point. So Mr. Tubman.
Yes, thank you. I really appreciated the presentation. On the blinded crossover design, so I understand this, basically you solve the problem of maintaining blindness. You don't have to unblind, but you do lose placebo. That group, the group that's been put into that blinded crossover system, they won't have a placebo group anyway. They more to care for purposes of.
That's right. You lose the contemporaneous placebo. That's right. You retain the value of having had a placebo at the beginning, though, because of the deferral, and that if there's a waning, particularly on the issue of waning immunity, obviously that difference might continue over time. So you retain some of the value of having a placebo at the beginning, but not all of the value, because things like particularly safety and other issues which might not be contingent just on the specific timing of when you got the vaccine, you will probably lose that or diminish that. You don't completely lose it. It depends on the characteristics of what it is you're measuring. There are many things we can look at still with this design, but that's why it has to be these designs coupled with the observational designs outside, which is obviously both what Pfizer has proposed, but also what the CDC and the FDA are also doing. So there's many things that can be learned outside the trial that we might have been looking to the trial to provide. Now will be depending more on observational designs if we move more to these trials. So we don't lose all the benefits of the placebo group, but we do lose some of them. but that may not be a practical option, which was my point.
Your position is that we should not move people into the blind crossover design until they're eligible in their particular societal group for getting it anyway. I will say that we have gotten a lot of comments from folks who are in trials, like dozens of them, and their line seems to be, well, if EUA is granted, I should be offered it. I'm not asking to opine in that, but my question is, your view is you should wait till it decidedly their group comes up, but if EUA didn't cover them at all, because that is one of the possibilities we might talk about later, would that help in the ethical determination that, yes, they should definitely wait, because after all, EUA has not been granted for their group yet anyway?
I haven't thought through that whole chain. All I will say is that they don't, again, I mentioned this previously, I don't think they are, it wasn't part of the initial contract. They understood that they could do without the vaccine for much longer, in fact, than they probably will have under a deferred policy. And they still get priority under this plan. It's just they get it first within their own priority group. So it's not that they are not owed anything. There is a reciprocity here in that when their, in a sense, turn comes up, they get it first through the trial mechanism. And so they're not completely, they're not disadvantaged by being in the trial. In fact, they're advantaged, just maybe not as much as they might want by demanding it today. But as I said, I don't think the ethical calculus is such, and other groups have not thought it such, that they are necessarily owed that just by virtue of being in the trial. But they do get a benefit.
Okay, thank you very much. I know these are very difficult situations and scenarios that we will need to be talking about again during the first part of our discussion. I apologize also to those people who I haven't been able to call on. You'll have your chance during the discussion because there's going to be a lot that we're going to have to talk about in the first part of our 2-hour period that we have assigned this afternoon. I am not sure, Prada, when should we come back? Because I know some of the public presenters are ready to go at noon Eastern.
Prabha,
Or
Kathleen, are you there? So we're scheduled to come back at, we're originally scheduled to come back around 12 o'clock, but we still have to call in all the public speakers. So let's just schedule it for right now, as long as Prabha, are you there?
Yes, I am here. And
okay, go ahead.
So, yeah, so we are scheduled to start at noon time. So are they all lined up now to go at noon, or do we have 5 minutes or extra to give them?
No, I need to call them in all now. So let's just, we will project to be scheduled for a 25 to 30 minute break, but we have to confirm all the OPH speakers come in. So let's, at this time, probably if you agree, we'll do a 25-minute break, but we may have to extend it a little to make sure we get time to get everybody in. Okay?
Yes. Okay.
Okay, that's the guidance I needed. Thank you. So we're going to try to resume around noon Eastern.
All right.
Thank you.
So thank you. So with that, we are going to take a break.
been answered, I think.
Hold on, Arnold. All right, and welcome back to the 162nd meeting of the Vaccines-Related Biological Product Advisory Committee break. I'd like to now hand it back over to Arnold. Arnold, are you ready?
I am. I'd like to welcome everybody to the open public hearing session. Please note that both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearings session of the advisory committee, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, its products, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of your travel, lodgings, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Prabha.
Good afternoon, everyone. This is Dr. Prabha Atreya. I'm going to conduct the open public hearing session, and I will read your names in the order, and then when I call your name, please unmute your phone and start speaking, and you have 3 minutes to complete your remarks. Thank you. The first name is Dr. Kermit Kubitz. To the vaccine advice. Hello, these are my comments to the Vaccine Advisory Committee on the BNT162b2 coronavirus vaccine. Cover efficacy, structured benefit, risk, and follow-up. I have no conflicts except for a lot of elderly relatives. Next slide. The Pfizer vaccine appears efficacious based on 95% relative reduction in infections and T-cell responses comparable to human convalescent sera. See Figure 2, page 22, and Figure 4, page 24. Some uncertainty is introduced by the possibility of mild asymptomatic infection, including among placebo patients, including feelings of pain or injection site effects that may have caused more vaccinated participants to be tested than placebo recipients. Next slide. Benefit risk. I have reviewed the available evidence, and as a Caltech graduate, despite the uncertainties, the structured benefit risk of Pfizer BNT162b2 is very positive. The coronavirus, as we have heard, is a serious medical condition with 3,000 daily deaths. While there are therapeutics, there are no prophylactics against COVID-19. Even in the event of approval of the Pfizer vaccine, limited supplies will require EUA of other efficacious vaccines when available, such as Moderna or others. Benefits of the vaccine include prevention of COVID-19, after the second dose, prevention of severe COVID-19, and preventing COVID-19 after the first dose. Risks appear to be mild injection site pain, fever, or fatigue. Increased reactogenicity for recipients over 55 is more than offset by the benefits of preventing COVID-19 cases in older adults with weaker immune systems. I note that allergy-susceptible persons should be informed of any issues in the EUA fact sheet following approval.
The net benefit is highly positive. I would recommend the vaccine to my siblings over 80 and my relative in an assisted living facility where there have been 3 cases for staff and 3 cases among residents. There is a need for follow-up of vaccination effects, including how long after immunization patients are protected, whether the vaccine prevents infections which cause further transmission, and how any violations of cold chain supply, dilution, shaking, or administration affect efficacy. Thank you, Dr. Fink, Dr. Gruber, Secretary Azar, Dr. Hahn, and Dr. Messonnier. They need this vaccine yesterday.
Okay, great. Thank you. The next speaker is Diana Zuckerman.
Yes, hi, can you hear me?
Yes.
I'm Dr. Diana Thank you. I'm Dr. Diana Zuckerman, President of the National Center for Health Research. Next slide. Our center scrutinizes the safety and effectiveness of medical products, and we don't accept funding from companies that make those products. My expertise is based on my postdoctoral training in epidemiology and as a faculty member and researcher at Vassar, Yale, and Harvard, and also previously a fellow in bioethics at Penn. I've also previously worked at HHS and the U.S. Congress. Next slide. Today I'm going to focus on 2 major concerns on how we can improve the data that are already available. Number 1, the 2-month median follow-up is just too short to have long-term safety information and long-term efficacy information, so it's essential that the randomized controlled trial be continued. Number 2, there's a lack of diversity in COVID cases. There were 0 Black cases in the vaccine group and only 7 Black cases in the placebo group, and there were 0 cases that were ages 75 and up in the vaccine group and 5 in the placebo group. And it was very disturbing. Wall Street Journal did a chart saying that it was 100% effective for Black patients, for example. So that's one of the reasons why we need more people so we have reasonable data. Next slide. There are also too few severe cases to draw any conclusions. There were only 4 severe cases after the 2nd dose. That's just too few to conclude anything. Next slide. Long-term patients were not, long-term care patients were not in the study. There were about 800 people ages 75 and over in the study, but only 5 were cases. So we want to save their lives, but how can we ensure informed consent to nursing home patients when we have no data to provide? And how many frail elderly or their family members can make an informed decision based on so little information? Next slide. We need long-term data to fully understand if the benefits outweigh the risks for frail patients and for all races and ethnicities and for all patients. And that's why continuing the randomized controlled trial is so important. Next slide. In conclusion, the EUA is not approval, and it should have more restrictions than you'd have for approval. So FDA should require continuation of the RCT while targeting EUA to priority populations, especially healthcare workers. I agree. Don't let anyone in the placebo group jump the queue. Second, EUA should not allow off-label use for non-priority groups, whether they're celebrities or anybody else. Off-label use could potentially occur under FDA's expanded access program. And last, FDA should delay access to vaccines by the placebo group unless they are in the priority populations. And I am concerned that the blinded crossover would be would be informative if the vaccine is not long-term efficacious. But if it was efficacious long-term, we would lose that information in a blinded crossover. Thank you very much for the opportunity to speak today.
Great. Next speaker is Peter Doshi.
Hi, this is Peter Doshi. Hopefully you can hear me. Thanks for the chance to speak. For identification purposes, I'm on the faculty at the University of Maryland and a medical journal editor with the BMJ. I have no relevant conflicts of interest and no one's paid for my attendance. Slide 2, please. My experience has been that careful review of a large trial takes considerable time and effort. As FDA has already reviewed the data, I'd like to know whether FDA is confident in the data collection for the primary endpoint, specifically that any unofficial unblinding did not affect the results, and that fever and pain medications did not mask symptoms, thus preventing case detection. I didn't find answers to these questions in the FDA's briefing documents. Slide 3. The dramatic difference in rate of side effects between vaccine and placebo raises questions about how well these trials could be observer-blinded. With a subjective endpoint like symptomatic COVID, blinding is important, but it seems fair to think that people could make reasonable guesses as to which group they were in. Slide 4, please. In the real world, the mantra has been to test, 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 but this wasn't the case in the trial. The study protocol says in the 7 days after vaccination, do not test unless, in the investigator's opinion, the clinical picture suggests COVID rather than vaccine side effects. This basically amounts to asking investigators to make guesses as to which intervention group patients were in. My question is, was this kind of judgment ever applied in the days it could affect the primary endpoint? And now I'd like to skip to slide 6 in the interest of time. Possible unblinding would matter less if the trials had been designed to directly test the vaccine's ability to reduce deaths, ICU use, and hospitalizations, as most people assumed the trials were set up to do. So it's great when the data look encouraging, but trials should be directly testing the endpoints that matter. Then there's the duration of protection issue. A vaccine that delivers a 95% relative risk reduction of COVID 2 to 3 months after vaccination is one thing. But for the many people who lack natural immunity and don't get exposed to the virus soon after vaccination, protection needs to last much longer. After 6 months or a year, will the vaccine still meet the FDA's 50% effective requirement? The trials just don't have sufficient data to say. Keeping the trials going with placebo-controlled follow-up will help answer the many crucial questions that remain. For those that do not wish to wait for clear evidence that benefits outweigh risks, an expanded access program can be set up. Access doesn't require authorization. And I just want to end by saying that whatever FDA ultimately does, the full trial data must be made publicly available. Thanks for offering me the time to speak.
Okay, the next speaker is Rasi Hassad.
Thank you. Thank you. I am Rafi Hassad, a professor at Mercy College with expertise in epidemiology, statistics, and mental health. I hereby declare no known or potential conflict of interest. Next slide. This vaccine is reported to have an impressive overall efficacy rate of 95%, consistent across key demographics. However, the primary efficacy endpoint of the trial was symptomatic COVID-19 infection. Participants who may have developed asymptomatic infection were not identified in this trial. Therefore, if as expected, an emergency use authorization is issued, the spectrum of COVID-19 infection that this vaccine is efficacious against must be emphasized. Next slide. Adequate statistical power is necessary for differentiating between an actual vaccine effect and one that occurred by chance. The power of this trial was calculated at 90% to detect an overall effect for vaccine efficacy and may not allow for firm conclusions regarding subgroup differences. Next slide. As reported, there were no serious safety concerns, which is quite reassuring. However, there were frequent mild and moderate adverse effects, which are recognized as common side effects of vaccines. Next slide. Additionally, participants were blind as to whether they received the vaccine or placebo. But given the common awareness of vaccine side effects, unintentional unblinding may have occurred, and this can potentially inflate estimates of vaccine efficacy. Next slide. In conclusion, the potential benefits of this vaccine
outweigh the identified risks. Therefore, I support the issuance of an EUA with the stipulation that the vaccine can protect against symptomatic disease. But at this time, it is not known if it prevents infections and transmission. Contraindication for those with a history of severe allergic reactions is plausible and should be included in the EUA as well. The continuing need for non-pharmaceutical interventions, particularly mask wearing, should also be noted. Finally, ongoing monitoring of this vaccine is imperative. Thank you.
Okay, the next speaker is Yvonne Fine.
Hi, my name is Evan Fine, and I'm a phase 1 participant in the Pfizer and BioNTech vaccine trial that was conducted by researchers at NYU. I have no financial or other conflicts of interest to disclose. I'm speaking today because after conversations with friends, family, and coworkers, I thought it was important to share my experience in this trial and to do my part to help ease people's fears about the vaccine. I think I got a real vaccine and not a placebo because of mild adverse effects such as fever, chills, and pain in the injection site after the second injection. After reporting the side effects, I was called repeatedly by the doctors and researchers at NYU to see if I was okay, and I was. Nothing felt rushed, and I never felt like a guinea pig. The question that everyone asks me is, well, what happened in the long term? Are there any long-term side effects? And it's been more than 5 months now since my first shot, and I can happily report that there are none. Since I participated in the trial, I've helped out my older parents, I've gone to work in person, and I've exercised in small groups, and I haven't gotten COVID-19. I also want to emphasize that these activities do not represent changes of behavior that would alter the integrity of the clinical data. These are life activities that I have to do anyway. Not all of us are able to lock down and stay at home indefinitely. I'd still do my best to follow health rules and safety precautions. I understand the concern about whether or not people will trust the vaccine, but there will always be some holdouts. Most Americans will take the vaccine voluntarily as long as we're honest, don't talk down to them, and treat them like autonomous adults. But right now, demand for the vaccine exceeds the supply. The skepticism of some does not justify delays for others who desperately want to take it. The best way to get people to take the vaccine is to lead by example. I don't think that the problem was that this vaccine was rushed. The issue is that other important innovations are slowed down too much by delays in scheduling, lack of funding, and other bureaucratic rules that do little to enhance safety protocols. If Pfizer, BioNTech, Moderna, or anyone else has an RNA product that can immediately help people with cancer, dementia, or AIDS, the FDA should accept the data on a rolling basis and work with these companies to get to and complete phase 3 clinical trials within a year or two. 5 to 10 years for these trials is simply not good enough. Pfizer has just set the gold standard for future clinical trials. Let's live up to this. Now, this is an emergency, and the burden of proof is on those who don't want to authorize the vaccine. Absent compelling reason not to authorize it, it is simply immoral and unethical to deny the vaccine to healthcare workers or first responders who want it. An EUA must be granted, and it must be granted tonight. As of yesterday, the daily death toll for COVID-19 exceeds the death toll on September 11, 2001. Delays on December 10th will mean more death on January 10th. In the words of Todd Beamer, let's roll. Thank you.
Thank you. The next speaker is Angela Rasmussen.
Thank you for allowing me a few moments to speak. I'm a virologist and affiliate at the Georgetown Center for Global Health Science and Security. I'm also, I have an advisory relationship with Siemens, but I have not been compensated to appear today. I'd like to offer some comments on how serology testing might better inform our knowledge of how these vaccines are working after they are rolled out to the public.
So this expedited process has moved considerably faster than the typical vaccine development timeline, and as such, we are inherently limited on the breadth and scope of data that's available on duration, durability, and effectiveness of the immunity, as well as the variability of different vaccinees' respective immune responses. Due to exclusion criteria in the clinical trial itself, there's also limited data available on minority and underserved populations, as well as pediatric populations, pregnant women, and the elderly. And we don't really know a lot about how these broader populations will develop antibodies to the vaccines. With the limited supply of vaccines, at least initially, it's going to be really critical to determine who has already been infected with SARS coronavirus 2 in order to conserve the vaccine for equitable access for others in the same priority category. Because this rollout will be so highly visible and pivotal, we really need to address potential problems such as outbreaks in nursing homes, hospitals, schools, or other at-risk populations due to a failure to seroconvert. And serology testing could really help limit all of these issues. Next slide, please. Serology testing could help at different phases also before and after vaccination. So prior to vaccination, serology testing could assist in prioritizing individuals for vaccination, as I just mentioned, also establishing a serological baseline in the population, and help ensure that a scarce supply of vaccines initially reaches the most vulnerable people. Shortly after vaccination, it can confirm initial neutralizing antibody responses to the vaccine. It can also help ensure that antibody response clears the threshold for protective immunity. And over the longer term, 3, 6, and 9 months after vaccination, it can confirm the persistence and duration of immunity. It can also provide the means of bridging bridge trials to additional populations and subgroups. Finally, it can assess the protective efficacy of vaccination by distinguishing antibodies from vaccination versus antibodies from natural infection. So by looking for anti-nucleocapsid antibodies, you could actually determine if people who have been vaccinated have been infected after being vaccinated. And finally, annually after vaccination, we can assess better the persistence and duration of immunity. It can also inform the requirements for future vaccinations that are developed on an accelerated timeline such as this one. Thank you so much for allowing me time to speak.
Thank you. The next speaker is Jared Krupnik.
Your next presenter. Great, thank you. Before I begin, your next presenter, Dr. David Berger, alerted me that he can't get back into the conference. If you could resolve that for him, so thank you. So slide number 1, please. My name is Jared Krupnik. I'm the president of Uniting for Action and the founder of the Vaccine Considerations Project. I don't have any conflicts of interest to share. Slide number 2, please. As you can see, the Vaccine Considerations Project has been created to highlight health and safety throughout the COVID-19 vaccine evaluation process. I want to acknowledge and thank the incredible team behind all this work. Thank you, Dr. Eric Brown, Dr. David Berger, and our incredibly talented, committed, and rapidly growing team of graduate students. Slide number 3, please. I don't have to tell you about the challenges this committee and this agency have been facing. You're living those challenges. I'm bringing up the challenges to highlight that amid all the different reasons for vaccine hesitancy, what remains universal is that widespread concerns must be adequately addressed in order to ensure sufficient buy-in by public health experts, medical professionals, and the public. Slide number 4, please. You're seeking to establish trust and confidence in any vaccine you authorize and in the evaluation process by being rigorous, comprehensive, and transparent. There are limits to what can be addressed in a meeting. We're working hard to help you expand those limits. We're creating new ways to organize and share information. Here you can see how we've thoroughly analyzed the transcript of your previous meeting to extract specific concerns expressed by each individual committee member. We're providing the link to our website, vaccineconsiderations.com, where the full process transcript is available for anyone to see. Slide number 5, please. As you can see on this slide, our team has plotted specific concerns from specific individuals to develop a matrix of concerns. By plotting each concern as a row and placing each of the committee members in separate columns, you can use this matrix to quickly and easily see which members share concerns and which concerns have yet to be addressed or fully resolved. You can use this document as a living document where each member can continue to add and update their concerns offline, and the FDA staff and other committee members can address those concerns. By making this public, things won't fall through the cracks. This reassures the public and the professionals that are relying on you. The matrix of concerns is available to all at vaccineconsiderations.com. Slide number 6, please. We're using this spreadsheet as a model to demonstrate the concept, but a spreadsheet is limited. That's why we spent over a year programming a custom platform called Information for Action that provides this type of functionality at scale. Our team is reaching out to stakeholders all around the country, experts, organizations, and associations to build and organize the central repository of concerns. We want to invite and encourage this committee and the FDA staff to utilize these technological tools to effectively address concerns, build trust, and reduce hesitancies. If you appreciate what we've shared, we'd like to connect with you. Please reach out to us at team@vaccineconsiderations.com. Thank you. My team and I look forward to working with you.
Great. The next speaker is Mr. David Berger, Dr. David Berger.
Hello. Thank you for allowing me to speak today. My name is David Berger. I am a board-certified pediatrician. I have no conflicts of interest. I am one of the few pediatricians in Florida who does not discharge families from my practice if they have vaccine hesitancy or do not wish to follow the recommended CDC schedule. My comments today are very mindful of the strong overall benefit our nation will get when most of us have immunity to SARS-CoV-2. Next slide, please. My presentation today is about vaccine hesitancy and steps that can be taken to increase my confidence in the COVID vaccine program. And this should be slide 3, by the way. It is very important that we see ongoing complete transparency about the vaccine in terms of both safety and efficacy. We have seen reports that up to 50% of Americans and 30% of physicians have some level of hesitancy about COVID vaccines. We must allow for meaningful public scrutiny to build public confidence in the vaccine program. Next slide, slide 4. True informed consent has always been at the core of how I practice medicine. As doctors, we have a duty to inform our patients about the benefits and risks of any medical procedure. If a person feels she is not given sufficient information about a treatment, how can she provide informed consent? I also think it's important to respect people who have concerns about hesitancy about vaccines. I often find that hesitant families will proceed with vaccines if they don't feel that their concerns were blown off and minimized, but instead were respected and tended to. Slide 5. While there are many subpopulations that should be studied for increased chance of vaccine side effects, there is particular concern about those with pre-existing allergic, hyperinflammatory, and autoimmune conditions. The Pfizer data shows that 4 individuals who received the vaccine developed Bell's palsy, whereas there were no incidents in the placebo group. On the first day of England's vaccine program, their government had to tell people not to take the vaccine, the Pfizer vaccine, if they have a history of significant allergic reactions after 2 significant anaphylactoid reactions were seen in recipients. Slide 6. It will be difficult to quickly and fully track all 50 states to find the real frequency of adverse reactions. It is important we have a strong federal program to coordinate this. Please ensure a plan for this. Please provide data for us to know if people who have had COVID infection already have an increased or decreased risk of a vaccine reaction. Please provide comparative data between the different vaccine products to determine if any brand may have more or different reactions than other brands. Slide 7. I implore you to put in place a very long-term post-vaccine surveillance program. Many autoimmune and hyperinflammatory conditions often take a while to develop significant enough symptoms for a patient to seek medical help. Slide 8. It is also important to have a robust surveillance of COVID IgG antibodies so we will have a way of knowing if immunity is waning. We need to know if what is considered a protective antibody level for COVID-19, just like we have for other vaccine titers that can be commercially tested. Slide 9. If the FDA can increase our level of confidence, hesitancy will likely decline, and the COVID vaccine program will have a better chance of success. It will take many good people to defeat COVID-19, and I hope I can help you make a difference. Next slide. Thank you very much. I appreciate the time.
Okay, thank you, Dr. Berger. The next speaker is Sydney Wolf, Dr. Sydney Wolf, and he does not have PowerPoint.
Just like we have for other vaccine titers can be commercially tested. Slide 9. If the FDA can
Dr. Wolf, you can't listen to it on the... Dr. Wolf?
Yes.
Okay,
Can
Dr.
you hear
Wolf,
me
you
with
gotta
the
turn your,
speakerphone on?
no, yeah, you have to turn your TV off or otherwise we're going to hear the delay. So please turn your TV off or whatever you're listening to.
I will turn it off right now. It's off.
All right.
Can you hear me
Dr.
on
Wolf?
speakerphone? I'm doing
Yes, we can,
speakerphone
sir.
now.
Yes, we
Can
can,
you hear
sir.
that?
Go ahead. Yes, we can. Go ahead, sir.
I'm Dr. Sidney Wolf, founder and senior advisor of the Public Citizens Health Research Group. I have no conflicts. With the surging pandemic and interim efficacy and safety results made public 2 days ago, we now agree with the need for an EUA for Pfizer-BioNTech COVID vaccine. There's an important unresolved conflict, though. If an EUA is granted for widespread use, should the 19,000 participants in the trial who received a placebo be notified of this and be offered a vaccine by Pfizer, clearly encouraging them to stay in the trial? Similarly, should blinded vaccine recipients be told of their status, thereby encouraging continuation in the trial? What would you wish if you were subjects in the trial? As status uninformed trial participants, you might otherwise leave the trial to get vaccinated with the Pfizer or any other available vaccine. The unblinding vaccine providing proposal has important advantages. First, once an EUA is granted, the ethical obligation, as Pfizer says, to both inform all placebo recipients of their status and offer them vaccine within the context of the trial is met. Second, by retaining more trial participants than if the trial remains blinded after an EUA, more recipients may be followed afterwards. The originally vaccinated group could be compared with the newly vaccinated group to continually compare rates of new COVID-19 infection with increasing duration of vaccination as well as adverse reactions. FDA has said that it doesn't consider availability, as you have all heard, of a COVID-19 vaccine under an EUA in and of itself as grounds for immediately stopping a blinded follow-up in an ongoing trial or offering a vaccine to all placebo recipients. During the October 22nd meeting, during Dr. Dora Fink's presentation, someone asked about what about the problem of retaining placebo patients post-EUA, and Dr. Fink responded, with regards to mitigating the risk of dropout from ongoing clinical trials, we do share that concern. I don't have any specific remedies to offer at this time. We've asked the vaccine manufacturers to think carefully about how they would ensure clinical trial retention. Pfizer has stated in the briefing document, quote, it intends to continue the pivotal phase 3 study with participants in both the vaccine and placebo groups as originally allocated for as long as possible, emphasize that. Nevertheless, we have an ethical responsibility to inform all ongoing study participants of the availability and eligibility criteria of the vaccine made available under an EUA. They would accommodate those trial participants wishing to leave the trial by revealing which group they are in, but they can receive the vaccine only when practically eligible, depending on the government-specified priority group and the available supply. But Pfizer's real preference, and this is in the briefing documents, is that such placebo individuals are vaccinated within the study in order that both safety and efficacy data can continue to be collected. We believe this approach will minimize the number of current participants who withdraw from the study. If you were in the trial, would you prefer being unblinded only if you wish to leave the trial to seek possibly available but needed vaccine, or being automatically unblinded given Pfizer's vaccine if in the placebo group, or happy to find out
to wrap it up
to stay in the trial? Thank you.
Thank you. The next speaker is Kim Witzek.
Yes, Good afternoon. My name is Kim Witzek, and I'm speaking on behalf of Woody Matters, a drug safety organization started after the death of my husband, due to an undisclosed side effect of antidepressants. We represent the voice of families who live every day with the consequences of the current drug safety system. I am also on the board of directors for USA Patient Network, an independent patient voice advocating for safe, effective, and accessible medical treatments. Right now, the world is looking for hope so they can get back to normal. Too many lives and livelihoods have been lost. Like many of us, we put blind faith and hope in the system that ultimately failed us. I have several concerns about rushing novel vaccines to market. The public needs assurances that the FDA's review was thorough and independent, given the process for reviewing new products usually takes 6 to 10 months. How could the FDA be as rigorous in weeks this time? Is there a process in place if there are dissenting scientific reviews within the agency? This needs to be available, and FDA scientists need to be protected for whatever their judgment is and not be political like it has been with many of the past controversies such as antidepressants and suicide has been handled within the agency. Transparency is everything. Assuming FDA approval, then all trial data must be released and made public. This also includes the process information like Pfizer's Data Monitoring Committee, who determined it was safe. Is it public? If not, it should be. We also need a transparent process for catching safety signals and communicating with the public. It needs to be real time like we saw in the UK yesterday with the allergic reactions, and not reported in weeks, months, years later like history has shown. Post-market monitoring will be more important than ever. We really don't know what short and long-term harms are given the short duration. Will FDA be staffed to handle the large task of closely monitoring that will be needed? Like MAUDE, the medical device reporting system, which includes patient narratives in the reporting, the FDA needs to do the same for the VAERS. Narratives can help tell a more complete story than just the data without giving away important patient details. Finally, I have huge concerns with possible unblinding of placebo participants and giving them the actual vaccine, as Pfizer CEO alluded to be willing to do. If this happens, we lose our control group. In closing, I'm directing this comment to the news media. You have a huge responsibility to dig deeper, ask critical questions, and not be just an extension of the manufacturer's PR department and accept press releases like the Wall Street Journal article did earlier this week and included a Pfizer supplied chart showing efficacy by subgroups where blacks had 100% efficacy. This should have sounded alarms and begged for additional questions, and then they would have dug deeper and realized there weren't a whole lot of blacks in the trial. Please seek out independent researchers, scientists, and others without political or financial agendas. Ultimately, the public is the real-world clinical trial. It is one big human experiment. The only ones that have 100% immunity in this will be the pharmaceutical companies. They get all the benefits of sales without any of the legal liability should something go wrong. I'd like to thank you for your careful consideration of my comments. I know firsthand the importance of your advisory committee work. Thank you.
Okay, the next speaker is Ms. Ligda D.
Hi, thank you. I'm from AIDS Action Baltimore and the AIDS Treatment Activist Coalition. I have no conflicts of interest. The answer to both agency questions on COVID-19 prevention and the risk-benefit ratio are clearly yes, in my opinion. Many of the issues submitted in my written comments have been addressed in the sponsor submission. One important ethical issue involving maintaining participants on the placebo arm after EUA has been discussed. I think it is very reassuring that the sponsor is proposing that the placebo participants be permitted to decide whether to remain on the placebo or to receive the vaccine within the study at the appropriate time. The crossover design will hopefully ensure that the study will not crash. Crossovers are nobody's favorite, but an unblinded crossover is probably much more feasible here. Lay people have no understanding of the nuances discussed today. Acceptable designs that will confirm initial EUA data and protect current participants and suspicious future potential participants promoting public trust are essential. I do have a few important remaining concerns. The DART study for pregnant women should be much further along by now. Also, the sponsor is essentially claiming that safety and efficacy have been established across race and ethnicity. The analysis of 3,800 phase 2-3 participants includes only 0.5% Native Americans, 9.3 Black or African Americans, and 28 Latinx people. The Native American and Black or African American numbers are especially concerning in these key populations who are disproportionately affected by and who experience greater adverse COVID-19 related comorbidities and death. While my community appreciates the eventual inclusion of people with HIV, HBV, and HCV per Amendment 6, the mere 120 people with HIV enrolled is abysmal. The sponsor has submitted 0 separate data on people with HIV, HBV, and HCV, or people over 75. Thus, there is absolutely no data to guide vaccine use in these populations. This is especially concerning for people with HIV, as there is mounting evidence of disparate adverse co-infection outcomes. VRBPAC should recommend that the sponsor conduct post-EUA research to establish safety and efficacy in significant numbers of people over 75, in people of color, and people with HIV, HBV, and HCV where feasible, without excluding them from any indication. Finally, I hope we don't begin by lagging behind in the enrollment of people in color in COVID-19 research. I sincerely hope Pfizer will include consumers in its vaccine expert safety subcommittee and will convene community meetings to discuss trial designs as well as vaccine education and accrual and retention issues. This practice has served both the HIV community and sponsors very well over many years of successful and mutually beneficial drug development. Thank you and the FDA for such dedicated service and for the opportunity to comment.
Great, thank you. Next speaker is Peter Lurie.
Good afternoon. I'm Peter Lurie, President of the Nonprofit Center for Science in the Public Interest and an Associate Commissioner at FDA from 2014 to 17. I have no conflicts of interest to disclose. I would like to thank the FDA for conducting its review in a transparent manner and for committing to the advisory committee review process, particularly under concerted political pressure. I just want to address 2 issues today. First, based on the data accrued to date, the Pfizer product demonstrates a striking degree of efficacy in preventing confirmed COVID-19, one that is shared across a variety of demographic, clinical, and other subgroups. Now, I agree with the FDA reviewers that there is no evidence of a major safety signal. However, the extent of more minor adverse events is notable. These include injection site reactions, fatigue, headache, all in over 50% of subjects, and chills in almost a third, all substantially elevated compared to rates in the placebo group. I do not believe that these events should stand between this product and authorization, but I do think the rates of these events are sufficiently elevated to merit open and even-handed discussion with patients. We are already facing significant levels of vaccine hesitancy, and if patients are not forewarned about these adverse events, the word will surely spread rapidly, potentially exacerbating the hesitancy problem. I also welcome FDA's identification of the disproportionate numbers of Bell's palsy cases and hypersensitivity-related adverse events observed in the treated group as matters that should continue to be monitored, including in the post-marketing phase. The second issue relates to trial design now that at least one safe and effective vaccine has been identified. The issues are complex, but we ought to be able to agree on this. No subject who has put their body on the line in a vaccine study should be at a disadvantage in terms of vaccine access as a result of their participation. Yet some observers appear to be advocating for extended periods of blinded follow-up even after authorization. This position is hard to justify ethically if it is inconsistent with public health recommendations at the time, particularly with rapidly rising case rates and the reported levels of effectiveness for the Pfizer and Moderna vaccines. So let me propose the following framework, which I think we can all agree on, at least most of it. 1, subjects should be informed if any vaccine candidate is authorized, not just the one in their trial. 2, like any study subjects, those in vaccine trials should be given the opportunity to leave the trial at any time if they so desire. 3, given the shortages of available product, subjects should be offered vaccination with an authorized vaccine as soon as it is offered to those in their clinical or demographic group in accordance with federal or state guidelines. 4, those for whom the product is not yet recommended can continue to be followed in blinded fashion. And 5, vaccination is recommended for an individual. A good option is to do so in a blinded crossover manner as described by Dr. Goodman to facilitate blinded follow-up for long-term safety and efficacy outcomes. I believe that this will facilitate the collection of essential data while honoring the contributions of the tens of thousands of people whose altruistic efforts have brought us to where we are today. Thank you.
Okay, thank you. The next speaker is Andrew Spiegel.
Yes, thank you. Thank you for allowing me the opportunity to provide comments on this most important day. My name is Andrew Spiegel, and today I present in my role as chair of the World Patients Alliance. I have no disclosures. The World Patients Alliance is a global umbrella organization of nearly 200 patient organizations from 86 countries representing hundreds of millions of patients across diseases worldwide. My very brief comments today fall into three basic areas: dedication, commitment to science and transparency, and innovation. First, we want to applaud the dedication of the scientists, both within the private companies and within the FDA. In the last year, scientists and researchers have fought tirelessly to create a vaccine for the novel coronavirus and put it through rigorous processes to ensure its safety and its efficacy. We are comforted in knowing that the FDA will require rigorous post-distribution data monitoring to continue to ensure any approved vaccines will be safe for the population. We know that in these unprecedented times, the people of America and the people of the world deserve to know that their governments are working to eliminate this threat. And we want to make sure that the FDA knows that we will continue to hope that it keeps the patients at the forefront of all of its policy and approval decisions on these vaccines as well as other medicines. The entire world is watching what the FDA is doing, and as everyone knows, they're relying upon the FDA's commitment to patients. Last, as the coronavirus vaccines continue to be approved and used in countries around the world, we must continue the process of innovation. Developing countries need access to innovative medicines now more than ever. In future breakthroughs, the vaccine for COVID could be manufactured as a pill that would be easily distributed and prescribed in those countries with less access to healthcare professionals around the globe. I certainly am in agreement with other speakers before me on the issue of ensuring diversity in clinical trials and ensuring trial members get access to the vaccine, and will not repeat further comments on that. Thanks again to all the stakeholders who have come together in an unprecedented way during these unprecedented times to do what would be unthinkable in the past. On behalf of patients around the globe, we thank you for your hard work and your commitment.
Okay, great. The next speaker is Nisa Shafi.
Good afternoon. I'm Nisa Shafi, and I'm here today on behalf of the National Consumers League. I have no relevant conflicts of interest. We extend our gratitude to the Vaccines and Related Biological Products Advisory Committee for the opportunity to present public comment today on behalf of consumers regarding the deployment of an emergency use authorization for the Pfizer BioNTech COVID-19 vaccine. Firstly, we want to thank the Food and Drug Administration, Centers for Disease Control and Prevention, and other public health agencies for their demonstrated commitment to fostering public trust throughout the COVID-19 vaccine development and approval process. As consumer advocates, we have been encouraged by the honesty, transparency, and access afforded to the public during this critical time. To that point, there has never been a more critical time for consumers to have confidence in the FDA. The agency has undergone scrutiny from the scientific community for prematurely issuing EUAs for COVID-19 therapeutics. NCL is aware that developing a vaccine for COVID-19 is a time-sensitive priority and appreciates that the FDA recognizes that an EUA is not intended to replace long-term randomized clinical trials data associated with full FDA approval. We look forward to continuing guidance around the vaccine as the trial continues to collect safety and efficacy data 2 years post-release. We have great trust in the FDA's rigorous vaccine approval process and call on the agency to perform ongoing post-market surveillance to ensure the vaccine's continued safety and efficacy. Post-market surveillance performed in the UK yielded that the vaccine is unsafe for individuals with severe allergies. We call on the FDA to heed these warnings and to continue to sustain its robust interagency collaboration towards the evaluation, approval, and distribution of the COVID-19 vaccine. Consumers will rely on ongoing guidance from public health agencies regarding any potential adverse events from the vaccine. Additionally, ensuring innovative vaccine delivery methods, such as including oral or nasal options, could address geographic access issues as well as adequately consider diverse health needs, increasing overall uptake. We welcome efforts to ensure diversity in the clinical trials for the COVID-19 vaccine, and NCL requests that the FDA continue to prioritize vaccine clinical trial data that reflects diversity, as people of color will need to have confidence in the vaccine's efficacy in their communities. This will impact overall uptake of the vaccine. The development of a COVID-19 vaccine in such record time has been a miraculous feat, made possible through robust collaboration between private and public entities. NCL will continue to support the FDA and CDC in its effort to release a COVID-19 vaccine safely and expeditiously. Thank you to the committee for your consideration for our views on this important public health issue.
Okay, thank you. The next speaker is Julie Omahundro.
Good afternoon. I'm Julie Omahundro, unaware of any conflicts of interest. I will cover key points in the comments I submitted and leave you to read through those as you please. They were submitted under the banner of the Regulatory Watch Cat and easily identified by a cute black cat. I start with a question. Does the EUA violate Article 37 of the Declaration of Helsinki, which states that unproven intervention should not be used in medical practice without informed consent? This led to the questions of whether authorized products are proven or unproven, and whether their use is medical practice or research. Eventually, I concluded that EUA lies in an ethical gray area, falling along several continuums. I considered a few such continuums and ethical questions that they might raise. I also looked at ethical standards in 21 CFR, the Belmont Report, and the AMA Code of Medical Ethics. Finally, I considered Section 564 of the Food, Drug, and Cosmetic Act and the current status of both informed and consent for EUA products. Section 564 requires informing patients that the product has been authorized for emergency use. The problem is that patients don't know what that means. Currently, providers and patients are provided with product fact sheets, all apparently based on the same template. The language addressing the product has been authorized for emergency use comes directly from Section 564, which was not written to inform patients of anything. It is not in language understandable to patients nor to providers. Currently, there is much confusion among manufacturers, providers, and patients regarding the regulatory status of authorized products and what can reasonably be expected of their safety and effectiveness. It is the latter that patients and providers need to understand and where the fact sheets fall far short. If individuals with the appropriate expertise could evaluate concepts such as proven versus unproven, medical practice versus research, as they apply to EUA and as points along a continuum, this could provide a rational framework for addressing ethical concerns that have emerged with the EUA, as well as the appropriate regulatory oversight and the appropriate use of clinical data generated from the use of an EUA product. I don't think this type of evaluation is suited for warp speed. Therefore, in the interim, I ask the committee to consider that the risks presented by the EUA include not only ethical risks, but also loss of public trust, a serious risk for any public health program. If issues emerge with the use of a new medical product, patients and providers rarely go back to, what was I told? I hope FDA will give serious consideration to moving forward with any COVID-19 vaccine initially under expanded access rather than EUA to assure adequate ethical oversight, informed consent, and early access to the vaccine by the populations that most urgently need it. In addition, for the love of Harvey Wiley and Jonas Salk, please, please have an IRB take a look at the template for the patient fact sheets. Thank you.
Okay, great. The next speaker is Drew West.
Hi. Thank you. Thank you for this opportunity to comment. My name is Drew West, and I'm the president of the USA Patient Network. Our organization is composed of many patients and family members who have been affected by pharmaceutical and medical devices that promised hope but left our members or family members harmed with lifelong side effects and sometimes death. We have no conflicts of interest to report as we are a completely independent voice for patients and their families. Our concerns are based on lived experience. Every person receiving a COVID vaccine needs to know the answers to basic questions, such as, will this vaccine prevent the most serious symptoms or the spread of the disease? What side effects or problems might occur immediately, midterm, and long-term? And how long will this vaccine protection last? It's our opinion that at this time there are too many unanswered questions regarding the safety and efficacy to release a vaccine. The release of these vaccines will make participants unwitting subjects in a seemingly uncontrolled clinical trial. That said, we recognize the tremendous pressure being placed on the FDA to act. Therefore, in the absence of reasonable assurances of safety and efficacy, we ask that the release of this vaccine be done cautiously and with thoughtfulness for the health and well-being of the recipients. We ask that you proceed slowly with release of these vaccines on a voluntary basis until full conclusion of blinded phase 3 clinical trials. We ask that you exclude populations that have not been studied or thoroughly studied, such as pregnant women, frail elderly persons, and severely immunocompromised persons, among others. We also ask that you continue or expand existing or new clinical trials to include groups of people who have not yet been included in any clinical trial to truly determine safety and efficacy. We ask that you monitor the vaccine recipients long-term over time by collecting and analyzing data to identify the real-world safety and adverse response results. This includes giving clear instructions and encouragement to vaccine recipients on what, where, and how to report adverse response events, not just reporting to their doctors. We ask that you give complete, informed consent information that includes full disclosure of the ingredients in the vaccines, all possible adverse responses, as well as a statement in plain language that the current safety and efficacy information is incomplete and that the vaccines have not yet been studied for safety and effectiveness for everyone or studied for adverse responses or effectiveness over time. We thank you for hearing our concerns.
Okay, great. Thank you. The next speaker is Sarah Christopherson.
Hi, thank you. My name is Sarah Christopherson. I am the Policy Advocacy Director at the National Women's Health Network. We're a nonprofit advocacy organization that has been bringing the voices of women to the FDA for 45 years. We are supported by our members and do not accept financial support from drug or device makers, and I have no conflicts of interest to disclose. We applaud the FDA for their diligent work during this public health emergency on desperately needed vaccines, including, yes, over Thanksgiving. However, we do have serious concerns that moving forward with an emergency use authorization based on so little data for so many of the communities that have been hardest hit by this virus will do little to assuage legitimate concerns in those communities about taking the vaccine. As we heard this morning, CDC data indicate that Black and Indigenous people living in the U.S. are roughly 4 times more likely to be hospitalized from COVID-19 and roughly 3 times more likely to die from the virus than their white counterparts. And as we heard at the October 22nd meeting, members of those communities have also expressed a really strong interest in knowing that the vaccine will work in people like them. Without those assurances, Black Americans have expressed high rates of COVID vaccine hesitation, and for good reason. Black Americans don't have to look back to the last century or to the infamous Tuskegee study to see examples of the medical system undervaluing them. And many Black and Indigenous people have had multiple discriminatory and negative experiences with the medical system in their own lives. Pew Research found a clear link between confidence in the regulatory process that you all are pursuing and Americans' willingness to get the vaccine. Before authorization is granted, affected communities need to have confidence that the vaccine is safe and effective for people like them. The efficacy data submitted to this panel included fewer than 2,000 Black or African American vaccine recipients and just 131 American Indian or Alaska Native recipients, not counting the placebo control population. The safety data included just 206 Black seniors, 65 or older, and just 131 American Indian or Alaska Native people of any age. If this advisory committee votes to recommend authorization based on this data, FDA must ensure robust tracking and bold public transparency once the vaccine is taken. Otherwise, the near-term side effects of the vaccine, which have been mentioned earlier, such as fever, chills, pain, fatigue, all of which could be really quite alarming to the public not expecting it, could further fuel distrust among communities that have been given little reason to trust this process. Thank you.
Okay, thank you. The next speaker is Vicky Pepsworth.
My name is Dr. Vicki Pebsworth. I have no financial conflicts. I'm a public health scientist and nurse who has served as consumer representative on VRBPAC. I am the volunteer director of research and patient safety for the National Vaccine Information Center and the mother of a child injured by his 15-month well baby shots in 1998. The normal U.S. vaccine development, testing, and licensing process takes 10 to 20 years. When accelerated and when populations studied are not large enough or do not closely match those targeted to be vaccinated, the public assumes unknown and potentially increased risks. Under the emergency use authorization, COVID-19 vaccines can be approved but remain experimental and unlicensed, while manufacturers are shielded from liability if the vaccines cause harm. The EUA fact sheet prepared for the public must be transparent and fully disclose known and unknown risks of the Pfizer-BioNTech COVID-19 vaccine to guarantee fully informed medical decision-making and to ensure public confidence. Specifically, the EUA fact sheet should disclose whether the vaccine is at least 94% effective in preventing SARS-CoV-2 infection and transmission, or only prevents severe COVID-19 disease, hospitalization, and death. Whether there is evidence for antibody-acquired and T-cell mediated immunity and how long it lasts. Whether there is a risk of enhanced COVID-19 disease when vaccine recipients are exposed to the SARS-CoV-2 virus or have previously received flu shots or other vaccines. Whether there is evidence for immediate and/or delayed serious vaccine reactions and poor health outcomes, especially in those who have already had COVID-19 disease, have severe allergies, experience severe reactions to previous vaccinations, or are chronically ill. The EUA fact sheet must clearly identify populations excluded from clinical trials. Over 40 criteria excluded certain people from some trials, including pregnant women and children, those with high blood pressure, obesity, diabetes, asthma, heart, lung, and kidney disease, neuro and immune problems, and those using certain medications or with a history of vaccine reactions or over age 85. Will the vaccine be safe and effective for them? The EUA fact sheet must clearly list all vaccine ingredients and disclose that an aborted fetal cell line was used to test the vaccine, as reported in a September 2020 paper published by 61 Pfizer and BioNTech scientists. By law, the EUA fact sheet must state that the consumer has the option to accept or refuse the vaccine. It is the position of the National Vaccine Information Center that using coercion and sanctions to persuade adults to take an experimental vaccine or give it to their children is unethical and unlawful. Thank you for your consideration.
Okay, thank you. The next speaker is Martha Nolan.
Good afternoon. Thank you for the opportunity to speak with you today. I am Martha Nolan, Senior Policy Advisor at Healthy Women. Healthy Women is the nation's leading nonprofit health organization representing more than 18 million women. We provide consumers and healthcare providers with accurate, evidence-based information about diseases and conditions, innovations in research and science, and changes in policy that affect women's access to treatment and care. I'd like to first thank you for the thoughtful and important work you are doing today and all that you've been doing throughout the pandemic. The discussion today is truly historic, and I commend all of you for your leadership and careful consideration of the data that is before you. Whether today or in the future, as COVID-19 vaccines are authorized by this body, I know that we are all cognizant of the challenges that lie ahead in terms of public trust. The science being presented today is truly remarkable, and the transparency of this meeting and the sharing of information from all of the companies as they work to develop COVID-19 vaccine candidates is the first step in bolstering public trust. As you're aware, recent surveys among the general public, but also somewhat surprising among healthcare workers, reveal concerns around the safety of COVID vaccines and many myths and misconceptions. When you look at communities of color, the level of skepticisms are even higher. All of us who work in public health must look to address this issue, and working together will be particularly critical. That is why Healthy Women is co-leading an effort with more than 60 other public health organizations representing patients, caregivers, and families, diverse communities, healthcare workers, older Americans, veterans, frontline workers, and scientists who work together to alleviate the concerns and hesitancy associated with the COVID vaccines. The COVID-19 Vaccine Education and Equities Project's missions is to educate and raise awareness of the importance of the COVID-19 vaccination for public health, the economy, and the broader society, as well as leading the conversation to ensure equitable access to authorized and approved vaccinations. The key to getting our lives back to normal here in the U.S. and across the globe hinge on the success of COVID-19 vaccinations. But we also know that vaccines are only as effective if they are trusted and taken by a majority of the population. We look forward to continuing our work to help build that trust, and thank all of you again for the work you're doing to ensure that the coming vaccines are safe and effective. Thank you.
Okay, great. Thank you so much. The next speaker is Mitchell Warren.
Thank you so much. My name is Mitchell Warren, and I'm the Executive Director of AVAC, a nonprofit organization founded in 1995 to accelerate the ethical development and global delivery of HIV vaccines and other new prevention options. And in March, we joined with several organizations to establish the Global COVID Advocates Advisory Board. I have no conflicts to declare, and we accept no funding from pharmaceutical companies. AVAC enthusiastically welcomes the safety and efficacy data of the Pfizer-BioNTech mRNA vaccine candidate being presented today. This is terrific news, to be sure, and a triumph of science and partnership. While the data to us do clearly warrant an EUA, they also require the maximization of additional data collection to address a number of remaining questions and issues that need to be addressed. One, the critical importance of distinguishing between an EUA and licensure under a BLA, and for you all to help ensure that continued data collection and a clearly articulated pathway and timeline for a BLA is provided. Two, there continues enormous need for the inclusion of diverse populations in COVID vaccine trials generally, and the data under review today provide limited information about the safety and efficacy data in diverse populations, including people living with HIV, other immunocompromised people, and those who are pregnant and breastfeeding. It is essential that specific requirements and timelines be articulated so that these key populations are not left behind. And I want to underscore my colleague Linda D's earlier comments on the critical importance of equity, diversity, and inclusion in research and review. With only 2 months of follow-up data as per EUA guidance, and it is essential that future BLAs include at least 6 months of follow-up. Therefore, continued blinded follow-up of the trial is warranted, while recognizing that some trial participants will want to exercise their right to leave the trial and if from the placebo group seek vaccination. If an EUA is granted, it will be urgent for the FDA and the companies to rapidly develop clear information, including an explicit reconsent process to outline the benefits, risks, and rights of maintaining in the blinded trial for both public health and personal benefit, and strongly encourage the committee to endorse the deferred blinded crossover design proposed by NIAID and presented earlier. In addition, we encourage the FDA to urgently issue guidance to other developers who will need to address this issue. Fourth, perhaps the biggest unknowns remain the durability of vaccine efficacy and whether the vaccine prevents asymptomatic disease and will limit transmission. A clear plan to collect and communicate information to inform answers from within the ongoing trial, as well as in the design of additional trials, is essential and should be clearly articulated when any EUA is announced. and should be strategically linked to other vaccine developers to jointly identify correlates, bridge data to other populations and platforms, and track use. As we've seen repeatedly in this pandemic and throughout HIV, clear evidence-based information is key. In conclusion, AVAX applauds this mRNA vaccine and both its VRBPAC and FDA for its commitment towards transparency, independence, and evidence-based scientific decision-making. This process is not just about authorization or approval. It is a beacon of independent review and transparency that will help to foster the trust necessary to rebuild confidence in vaccines, in science, and in our public institutions. Thank you for what you're doing today and throughout this process, and thank you for the opportunity to present, and looking forward to continued engagement with scientific and regulatory processes that move with speed of trust.
Okay, great. Thank you. And I think we are almost come to a closure. We tried the last person requested, and then we are unable to reach her out. So I think we can conclude because we already have past 10 minutes past the actual time for the next presentation. But before we go to sponsor presentation, the director of the Center for Biologics, Dr. Peter Marks, would like to make a few comments. Then we'll go to sponsor presentation. Thank you. Dr. Marks, you are on.
Thanks very much, Prabha. I just want to take a moment here to offer some thanks to various people. First of all, thanks to anyone from the public who's tuning in today. I think having this as a transparent process is very important as one of the steps we're taking to try to enhance vaccine confidence across the country. I also want to take a moment to thank all of the advisory committee members and the speakers today. Their contributions are immense, and we'll look forward to their discussion later on today. It's also very important for me to take a moment to thank a group of people at FDA that have put an incredible amount of effort into this advisory committee meeting while they're simultaneously getting ready for another advisory committee meeting and taking care of many other vaccines in various stages of development. The amount of work that has been put into this by those just setting up these meetings is tremendous, and I'd like to thank Prabha and all of the advisory committee staff, but also an incredible debt of gratitude to all of our reviewers in various offices, and especially in our Office of Vaccines Research and Review. The leadership and the staff in that office have worked tirelessly throughout the past weeks and months to get to this point, and that included working diligently over this past holiday when they could have been spending time with their families. So we're very, very grateful for all of the work and grateful to all of you. So with that, I want to let this proceeding move on since it's going to be a long day anyway, but thank you very much.
Thank you, Dr. Marks. Now, Dr. Manto, back to you. Take on from this point on for the next presentation with the sponsor.
Right. Next, we have the sponsor presentations. The moderator is Catherine Jansen, Senior Vice President and Head Vaccine Research and Development of Pfizer, and William Gruber, Senior Vice President, Vaccine Clinical Research and Development of Pfizer. You're on.
Good afternoon, members of the committee, FDA, and ladies and gentlemen in the audience. It is a real pleasure to be here today. I'm Dr. Catherine Jansen, and I'm a Senior Vice President and Head of Vaccine Research and Development for Pfizer. I would like to thank the FDA for organizing the VRBPAC and the VRBPAC chair and members for their time. Pfizer and our partner BioNTech are pleased to be here today to discuss our candidate mRNA COVID-19 vaccine program in the context of requesting an emergency use authorization. Our presentation today will follow this agenda. After I I give a brief introduction. Dr. William Gruber, Senior Vice President, Vaccine Clinical Research and Development, will review the development program for our vaccine, including non-clinical, clinical safety, and clinical efficacy data. After this, I will come back to review the benefit risk and provide conclusions for our presentation. We are also pleased to have a principal investigator with us to answer any study-related questions. Dr. Stephen Thomas, who is Professor of Medicine and Microbiology and Immunology, Chief Division of Infectious Diseases, and Director, Institute for Global Health and Translational Sciences from the State University of New York, Upstate Medical University. Our COVID-19 mRNA vaccine, called BNT162b2, has been developed for the following indication: prevention of COVID-19 in individuals 16 years of age and older. The dose level is 30 micrograms with 2 doses given 21 days apart. The vaccine presentation is a 5-dose, multi-dose vial that is preservative-free and stored frozen between -80 and -60 degrees Celsius until use. Since the beginning of the year, we have been facing a devastating situation with COVID-19, the disease caused by the new coronavirus SARS-CoV-2. Tens of millions of people have been infected globally, and over 1.5 million people have already died. No one is safe from this disease, and certain groups, such as healthcare workers and first responders, the elderly, and people with underlying diseases, are at particularly high risk. It is now becoming very clear, particularly with the recent increasing rates of COVID-19 globally and in the United States, as discussed earlier by Dr. Hall, that we need a safe and efficacious vaccine to stem this devastating pandemic while also deploying other prevention strategies, such as infection control, including mask wearing and social distancing. SARS-CoV-2, the virus that causes COVID-19, is a new beta coronavirus that emerged late in 2019 in China. Shown in red are the coronavirus glycoprotein spikes, called S for short. The spike protein is an important vaccine target or antigen, as it mediates first the specific binding of the virus to the ACE2 host cell receptor, and then the fusion of the viral envelope with the host cell membrane. By these actions, the virus enters into human cells where it replicates, then spreads to other people, and often causing illness in the infected individual. Data available from other coronaviruses, such as SARS and MERS, had established that antibodies to S can block the binding of the virus to cells and prevent the infection of the human cell. So we chose the S protein as our vaccine antigen. Specifically, we chose the form of the S protein that was engineered to lock in the perfusion conformation, which is thought to be optimal for eliciting functional virus-neutralizing antibodies. We chose BioNTech's mRNA vaccine platform for a number of reasons, and giving BioNTech's long experience with messenger RNA vaccines in the oncology space. mRNA vaccines are produced by cell-free production processes using chemically highly defined vaccine components, RNA and lipid. mRNA vaccines can induce broad immune responses, well suited to address a new pathogen in a situation where there's no or limited knowledge of what correlates with protection. The mRNA vaccine platform lacks unwanted viral or vaccine platform antigens that may lower a vaccine-induced immune response. As a consequence, mRNA vaccines can be boosted repeatedly, an important consideration when persistence of vaccine immunity is not yet known. Given that mRNA vaccines are essentially synthesized, they can be developed and scaled up quickly. A clear advantage over cell-based production processes when developing a vaccine in a pandemic setting. So how does an mRNA vaccine work? We have a deep scientific understanding of how such vaccines work. The mRNA codes, that means harbors the information for the vaccine antigen, which is a form of the S protein. The mRNA is stabilized to formulation with lipids to form what we call lipid nanoparticles, or LNPs. The LNP formulation is further optimized to allow efficient entry of the LNPs into human cells. This LNP is also optimized to be able to enter antigen-presenting cells that are efficient at stimulating a broad immune response, which we desired, given that we did not know and still do not know which response best correlates with protection. Inside the cell, the mRNA is released from the LNP. Just like any other messenger RNA in a cell, the mRNA from the vaccine can direct the synthesis of a protein by the cell's own production machinery. And since our vaccine codes for a form of the SARS-CoV-2 spike protein, that protein is also produced. Once produced, the whole S protein and its smaller fragments are presented to the human immune system that recognizes the protein as foreign, resulting in stimulation of T helper cells or CD4 T cells that activate other T cells and antibody-producing B cells. The antibodies can bind to the S protein on our SARS-CoV-2 and neutralize the virus, which means the virus is no longer capable of infecting a cell. Also produced are CD8 T cells that can eliminate virally infected cells. An important feature of mRNA vaccines is that they stimulate effective B cell and T cell memory responses. This ensures longer-term protection from viral infection and disease. An additional advantage of the mRNA platform is that the vaccine induces a Th1-biased CD4 positive T cell response that is linked to protection from viruses and thus minimizes a theoretical risk of vaccine-enhanced disease. The Th1 biased response is characterized by secretion of cytokines involved in fighting a viral infection, such as interferon gamma and IL-2. So in a nutshell, the immune responses induced by the mRNA vaccine are similar to what you may get in response to a viral infection. But, of course, the mRNA vaccine is non-infectious and cannot cause disease. When we started our COVID-19 vaccine partnership with BioNTech, we had the choice of a number of different mRNA vaccine candidates, different flavors of the mRNA, and different forms of the spike protein. Given the enormity of our mission, clinical data were important to us in deciding on the right candidate for a COVID-19 vaccine. So we evaluated not just one, but 4 different candidates in phase 1 to be able to make real-time scientific decisions to select the best candidate based on the following major selection criteria. With regard to safety, we were looking for the most favorable safety and tolerability profile in both younger and older adults. With regard to immunogenicity, we were looking for the broadest antiviral immune responses most likely associated with efficacy. And with regard to a rapid pandemic response, we were looking for the candidate that could be developed and produced most efficiently. Using these criteria, we selected BNT162b2, which contains a modified RNA that expresses a codon-optimized, full-length, pre-fusion stabilized spike protein of SARS-CoV-2. The top priorities for vaccine development in general and for COVID-19 vaccine in particular, are as follows. We must demonstrate that the vaccine is highly effective, meaning that it can help prevent COVID-19 in at least the majority of vaccinated people. We must demonstrate that the vaccine is safe with a robust safety data set generated in a very large pivotal efficacy study. And we also must demonstrate that we can consistently manufacture the vaccine to the highest standards. Throughout 2020, we have been asked often, how can this be done in a year or less, when the process normally takes many years? Well, vaccine development or anything else in a pandemic setting is not normal. It requires a completely different way of thinking and a strategy of parallel versus sequential R&D. Instead of waiting for preclinical and clinical data to emerge, substantial efforts and resources were poured into process development and manufacturing scale-up well before any clinical data were available. This has been an unprecedented investment early in development. In planning clinical development, we worked with the FDA and other regulators on a seamless trial which collapsed phases 1, 2, and 3 clinical development, also in an unprecedented way, with close to real-time communications and decision-making without ever stopping the trial. We are grateful to FDA and other regulatory agencies who have lent us their enormous support to work on these seamless development timelines. With substantial work on the mRNA platforms already done at BioNTech, we were able to quickly start clinical development and advance to phase 2/3 start at the end of July this year. And we had support from CDC to provide real data on regional SARS-CoV-2 and COVID-19 attack rates in the United States to optimize our selection of trial sites. Last, but certainly not least, with the dedication of investigators and site staff, as well as the motivation of tens of thousands of trial participants, we were able to enroll over 40,000 participants in a record 6 weeks, give them each 2 doses of vaccine, and ensure their safety while conducting the trial with strict compliance and uncompromising quality. And now, let me introduce for you the clear and compelling data package you will hear about today that we believe satisfy the FDA guidance for COVID-19 vaccine emergency use authorization. We have submitted all of the information required to meet the EUA guidance, and you will hear about them this morning. As for manufacturing data, FDA has reviewed the CMC data submitted to date for our vaccine and has determined that the CMC information is adequate to ensure the vaccine's quality and consistency for authorization of the product under an EUA. And now, Dr. Bill Gruber will discuss the non-clinical and clinical data sets with you, as well as our future plans.
Thank you, Catherine. It's my pleasure to share with you today the development program for our vaccine candidate, BNT162b2. I'm going to begin with a very brief summary of the non-clinical data that encouraged us to move forward into the clinic. This includes both toxicity studies as well as a study looking at a challenge model in rhesus macaques. These studies are described in the briefing document. 2 toxicity studies in rats, including the BNT162b2 vaccine construct, were completed with no safety concerns. Development and reproductive toxicity studies are ongoing, with preliminary results available by mid-December. In a SARS coronavirus
coronavirus 2 rhesus challenge model, the BNT162b2 construct provided complete protection in the lungs as determined by nucleic acid amplification testing for SARS coronavirus 2 in bronchoalveolar lavage fluid. This information is now published. And importantly, there was no radiologic or histopathologic evidence of vaccine-elicited disease enhancement. Despite limitations of animal models, these findings anticipated results in our phase 3 clinical trial in which there is no evidence of enhanced disease. These results were encouraging, satisfied FDA guidance criteria, and permitted progression of human clinical trials. I'm now going to share with you the clinical safety, immunogenicity, and efficacy data from our overall clinical development program. First, I will cover safety and immune response from the German and U.S. studies, and then cover aspects of the phase 2-3 trial finishing with safety and efficacy results. So let's begin with the 2 phase 1 studies. The German phase 1 dose-ranging study was conducted in individuals 18 to 55 years of age. 12 subjects received the active BNT162b2 vaccine for each dose level cohort. This study evaluated safety, binding and neutralizing antibody responses, as well as cell-mediated immune response to look at the potential for Th1 bias, CD4, and CD8 T-cell responses. The U.S. study is a seamless study where we had a phase 1 portion that moved into phase 2 and then phase 3.